groups may be a very good way to identify early biomarkers, and of course, new leads for drugs and repurposed drugs. Everything can come in handy with that. Thank you very much. And this talk was not really for the human genetics scientists here, but it was really for the rest of the intelligent laity from other disciplines. Thank you.
Thank you, Professor Thelma. Now I invite Dr. Shinjini Bhatnagar. She is a pediatrician by training and spent 25 years in AIMS, then moved to THSCI. She is well known for her cohort studies and also made a lot of intervention and management for childhood diarrhea. And her work in recently known is Dharmini cohort studies, and she will bring more of clinician's perspective for all of us.
Thank you. Thank you. Thank you for having me here. I'm not, at the outset, disclaimer, I'm not a geneticist. I do not know much genomics, but yes, I have learned as a pediatrician and in child health research over the last couple of decades. So I don't know, the slides keep going on and off, so I will talk. I hope you can put the two connects. Dr. Thelma has already alluded to some of the work on maternal child health in genomics. And may I have my slides, please? He's saying he doesn't have my slides.
Sorry?
He's saying he doesn't have my slides.
No, it's there. It is there. So just move to next, no?
Maybe he's got the order.
No. Check.
Yeah, yeah.
Should I go and check it?
You check? No, that's okay. Okay, so I'm taking up again. So the focus is on I'm just going to talk a little bit about a very vulnerable population, often very ignored, maternal and child health in that maternal and child vulnerable population. And one of the areas which has really changed and made a paradigm shift in management is in blood cancers. And everybody is aware now of in acute leukemias in children where there is tumor genotyping, leading to targeted therapy, and that has made a substantial, significant effect in management. And Dr. Thelma has already alluded to the newborn screening that is...
Forward or backward?
Makaru?
Yeah, backward.
This is forward.
This is forward.
Okay.
Yes,
so newborn screening, and the only thing I would like to add, and Dr. Madhulika Kabra is here, that there's still a long way to go there, and there is newborn screening. It's largely based on metabolites. The augmented genomic screening for newborn is not happening everywhere, not recommended at the moment in most of the policy documents, and is difficult to scale up for both cost as well as the way it is being done. But what is evolving is the polygenic risk scores for diseases. And a couple of examples are for childhood asthma and childhood obesity, which really is going to make a shift in management as we move forward. So taking that and giving you an example from one of the large programs sponsored by Department of Biotechnology, the maternal child health domain, on Garbhani, which is the Interdisciplinary Group for Advanced Research on Birth Outcomes, DBT India initiative. This is, today we have one of the largest cohorts in South Asia of more than 12,000 participants, of 12,000 mothers being followed up to look at risk stratification, early risk stratification for better clinical management for adverse birth outcomes. And I'm speaking on behalf of the team. It's not my Garbhani. It is our program where we are across Regional Center for Biotechnology, National Institute of Biomedical Genomics, Gurgaon Civil Hospital, coordinated by THSTI and other tertiary hospitals as shown on the slide. So what we found, and interestingly, and that's where the interdisciplinarity of the whole research comes in, and that is why it's important to consider that as the next step. And what we've seen is that when we looked at clinical and ultrasound image markers, the predictive accuracy of this was only 54% to actually predict or stratify mothers early for an adverse birth outcome like preterm birth, which for people who are not aware is one of the biggest killers at birth. And even those who are born 3 weeks early may have a lot of cognitive and learning disabilities as they move forward. So very important for a country like ours where it's almost 13% as seen from our own data real time. And therefore, when we looked at this, we were very disappointed with this 54% predictive accuracy. We used very refined methods as a next step using machine learning, Bayesian approaches. The predictive ability has only gone up by 10 to 12%. So now what the next step is to add the genomic markers and the genomic variants and the research that we've done to this, along with the proteomics and the metabolites, and our predictive accuracy is showing an improvement. And therefore, coming to the, very quickly, some of the data which has been led by Arindam at NIBMG and his team, we have, for the first time in South Asia, done the GWAS of over 6,000 mothers, which has given us a population and a trans-ethnic representation of genetic markers for preterm birth, also laid the platform for that early risk stratification can happen with this kind of research. Using that forward, and I'm sure Arindam will talk about it later in the day, we've now identified 66 SNPs which predict preterm birth. They are being converted into panels, and that's where the translation comes in. They are being, and we, after those panels are ready, we'll be validating them across different cohorts in the country before they can be implemented or taken for commercialization. In addition, we are also to understand the mechanisms behind this very complicated, complex disorders. They have looked at, we've found DNA methylation changes, early DNA methylation changes early in pregnancy, and also some very exciting work in the cellular markers at the placental level. So this would not have been possible if we were not an interdisciplinary group. And that, I feel, is one of it. You know, it will not stop at a repository of genes. It will not stop at the labs. We really need large groups working together to answer these questions, which can be applicable to the country at the population level and at the patient level. So I'm happy to answer more questions later. I had 5 minutes, so thank you.
Thank you, Dr. Shinjini, for giving clinician perspective. Now I invite Dr. Ramesh Sonti from International Center for Genetic Engineering and Biotechnology, Delhi. And Ramesh has been working in the field of plant pathogen interaction for very, very long time and known for this new variety of rice, and is going to bring us that how plant genomics is happening at society level.
Thank you, Tangraj, and thank you, Dr. Ninave, for inviting me here. It's a pleasure to be here with all of you. Now, our health depends not just on our genes and our environment, but also on the food that we eat. So today I will talk to you a little bit about how the revolution in genomics and associated technologies is leading to the development of better crops. There are four broad areas in which genomics has actually already had a significant impact on how we breed crops. And these are, I'll show you each one of them as we go by. One is called marker-assisted selection, which is a method of plant breeding where you use DNA testing to identify within a large population those plants that have the particular traits of interest. And this helps you speed up the process of plant breeding in such a way that you can get the plant or the variety of interest in a much faster, in a much more precise way, so that the crop can be given to farmers much faster. So the breeding cycle, which may have taken 8 to 10 years, is now down to 2 to 4 years. So that's the kind of impact this technology has had. First slide, please. Yeah, so I'll be talking about applications of genomics in agriculture, particularly relevant to crop improvement. Next slide. So as I said, marker-assisted selection, the way genomics helps marker-assisted selection is that the marker-assisted selection technology, or MAS as it is commonly called, depends on the availability of large numbers of DNA markers. When you have a lot of markers, then you can find markers that are associated with a trait of interest much more easily. So genome studies facilitate development of large numbers of DNA markers. And using MAS, as of February 2022, 74 novel varieties have been developed and released in India in 7 different crops. Now, that is in 2022. I'm sure this number has gone past 100 now, maybe closer to 150, and many more in the pipeline in India. And all over the world, you can imagine everywhere it is actually being done. So the kind of impact that this technology has had is tremendous, and we will see the impact on it as we move forward. This is just one example from our own work, which we did when I was in CCMB, Hyderabad, along with the Indian Institute of Rice Research, which is also located in Hyderabad. We took a rice variety called Samba Masuri, which was disease susceptible. It's susceptible to a bacterial disease. And using this marker-assisted selection, we developed an improved variety. And you can see what is on the bottom is green. That's the resistant one. And the one on top is disease susceptible, and it's actually dried up. It's a dried up field. So this is just one example. I mean, this has made a big impact for farmers who cultivate this variety in regions where this disease is present. But like that, there are 74 others that are already developed. And NIPGR, I think last year, they released a variety of chickpea that was also developed to marker-assisted selection that is drought tolerant and high yielding. So this is an example, and I'm sure that in the years to come, we will have many, many more such developments, that will impact farmers and consumers. Next slide. Now, one area that is coming up now is something that's called genomic selection. This is going to again have a much bigger impact in the years to come. And genomic selection uses machine learning and genotype data to predict varieties that have the trait of interest. Now, the simple traits, like as in humans, simple traits are easy to phenotype. In plants also, simple traits are easy to phenotype. Disease resistance, tolerance, that's easy to phenotype. But things like adaptation to climate change, where you require crops that are tolerant to drought, temperature, multiple stresses coming at one time. Now, the phenotyping is very hard to do. So we have in our gene banks, as I'll tell you a little bit later, we have in our gene banks large numbers of crop varieties that are stored there. we need to phenotype them. Many of them or some of them have these traits that we are looking for, but we need to phenotype them. But to phenotype thousands and thousands of crops for these complex traits is very difficult. So phenotyping is difficult, but with the advances in genomics, genotyping has become relatively straightforward and inexpensive. So the idea here is that you take your whole population, take a subset of it, which you would call the training population. Now that subset is going to be phenotyped and genotyped. So for a subset of your population, you have phenotype and genotype data, and then you use these genome selection models, which are basically dependent based on AI and big data. Now you go to the rest of the population. So you have a testing population on which you can base, that's the learning set. Then you use your testing population where you have only the genotypic data. And based on the genotypic data, you're going to predict which are the ones that have these traits for adaptation to climate stress that I was telling you about. So once you have them, you have the varieties, and then you can actually do mapping, which is again facilitated by many of these advances in genomics. Next slide, please. So as I said, our gene banks have hundreds, thousands. You know, the National Gene Bank at the National Research Center for, there is a genome center in ICMR and in New Delhi, which is actually, it's actually called the National Institute of Plant Genomic Resources. And it has 450,000 different accessions. And for rice alone, there are probably close to 70,000. For chickpea, maybe 20,000, and nidicrisat, another 10,000 is there. How do you actually valorize the traits of interest that are present in these? Again, genomics coupled with some phenotypic studies are being used to develop what are called core collections. And if you're interested, we can discuss that more. But the idea is to be able to efficiently identify within that particular set of germplasm that's there in our gene bank, those that have the trait of interest. Once you have identified them, the genomics tools have developed so well, genomics and bioinformatics tools, that it's pretty easy to actually map them. So this is example of the variation that's there in chickpea that's given my former colleague at NIPGR, Swaroop Parida. And then in rice, you can actually see. Now what DBT has done, recognizing the value of the germplasm that's present in our gene banks and how to get that particular traits that are there, how to valorize these traits that are in the gene banks. DBT has supported about 7 mission projects in 7 crops, including rice and chickpea, to harness the genetic diversity present in gene banks for crop improvement. And I think again, NIPGR has one of these projects, and I've been fortunate to be involved in, sort of monitoring how these programs go. And I can say that there's tremendous amount of new variation that's being discovered in these projects, which I think a few years from now will actually go into the plant breeding programs and translate into varieties. Last slide, please. Just one more. Yeah, so this is about genome editing. And as I said, food is not only the amount of food you make, you also have to have quality food. And this is the work that my colleague Dr. Tanushree Kaul has done, where she has taken a Samba Masuri rice variety and made it high iron and high zinc through genome editing of certain regulatory genes in rice. And you can actually see the light color in the wild type is light blue, it has less iron, and all these other lines that are genome edited have high amounts of iron and zinc also in them. So I'd be happy to talk to you more later. Thanks again for your attention.
Thank you, Dr. Ramesh. Now we heard about human genomics, plant genomics. Now let's hear about what is beyond genomics from Dr. Debashish. He's from NII, and his research interest for a very long time has been developing methods for understanding host pathogen interaction. He also uses different tools, particularly AI and ML tools for his own research. So let's hear about what is beyond genomics.
Yeah, so as a, yeah, sure, yeah. Good morning. So as a computational biologist, probably we won't be there without genomics because all the data comes from genomes and all this. But coming to this, this audience doesn't require any introduction about utility of genomics for society. But if you think from a country perspective, common man perspective, when did people get aware of the power of genomics? It is after COVID. From INSACOG, like we could sequence the things within the country, we could identify the variants, and even the wastewater monitoring came, and that is where, like all genomics has been there before, but it has been so complex to, so as Thelma highlighted, and this, it has been so complex to make people really aware of the utility of this. It will take time. It is not easy to this, but that is where it is, and that has encouraged a lot of other projects to come up, and everybody is looking forward to the data which is coming from these 10,000 genome projects or things like that. And there are other projects where we are directly associated is this one, like before I go to a role of genomics in immunology and other thing, is like this is the INTGS project. So Indian TB, the tuberculosis genome sequencing. So drug resistance TB or any MR and all has been a major challenge for the country. But the way you detect by phenotypic DST, it could easily take a couple of weeks to identify that this patient is for the resistance to this drug. But there are, as Professor Sonti said, there are distinct genomic signatures which are linked to phenotypes. And for bacterial genomes, it's not that difficult to find it. So now there are distinct genomic signatures where you can tell if you can do sequencing in the clinic, and with technology like nanopore and things like coming, and it is indeed not far where you can rightly do the sequencing in the clinic and tell that this is. So these are some examples like INTGS where like many of the DVT institutes are involved. ICGB, Dheeraj is there, Anis from NII, even CCMB and NIBMG, and all these institutes are involved. And like this Human Genome Network gave the chance these are the centers which work for INSACOG, lead hubs or things like that, and that network is being used for other types of genomic projects. And coming to, and since I am from Immunology Institute, like if you think of a complex area like immunology, and without genomics, like immune genetic polymorphism and all, is very much dependent on your T cell repertory and all these things, and integrating WGS with studies, but you can really decode the complex interplay. Like we all do, there are many groups in NII we are doing single cell transcriptomics, but you are all interpreting the data on a reference genome which comes from elsewhere. So ideally, the situation would be we could do it using a reference genome which is from our country or something. That is where, because it is the lack of data, we are not able to integrate genomics to this. Even if you think of proteomics, you do not directly identify a protein from the mass spec. You go to the genome, translate it to the protein, and then you search for those things. So many of the things you are doing, all multi-omics is key, is very much dependent on genomics, and that is what we are trying to look from this. So next thing what I go to, I'll just give you one example like this one. There was like we had the COVID genomics project, which was it was sequencing the virus, but there were international projects, is like host genome initiative, like whole genome sequencing of around 7,000 individuals were done to find out which are the markers of like high risk or people severe COVID or something. There are many interesting genes have been identified which are this one, but all this required, this is from a study from England, so that requires comparison to like a European ancestry data. And ideally, sir, what we would require to have it, probably it will be good if we can have reference genome of our own this thing. And coming to this, next what I will talk of some things which are a little beyond genomics or something. And all of this, I think when Thelma pointed out, there are errors in the spelling mistake. What she was alluding to is genome is like a language, and you are looking, and with this, if you think of AI/ML, large language modeling, and that is what actually it is going to do for the future. And all of the power of machine learning has been recognized is like the protein structure prediction. And within just 1 to 2 years of its discovery, if any like a science or technology has been recognized by a Nobel Prize, it is probably the protein structure prediction. And that is being increasingly used in all omics and all these things. And there are language models are also being used for structure prediction, like you have AlphaFold. There is something called ESMFold, which actually uses the language modeling. It is computationally fast and it has a lot of potential. And if you come to like in genomics, you will find many variants like experts like Thelma and all. Right now, the situation is, if you found a variant, you want to tell, is it pathogenic, it is benign, or it is, you will see, like, of course, the real way to do that is to go to ClinVar, where there is a well-annotated, curated data supported by clinical phenotypic, this one, then you can say, okay, this is associated with this is pathogenic. But that you can do only for a small number of variants. A large number of variants will be predicted by many of these things as variants of unknown significance or something. And this is where you can use the data, you can use your understanding of the biology, you can use your understanding of the protein structure to see, is this variants have any significance. In fact, after development of AlphaFold, you have a program called AlphaMissense, which has been developed by the same group, DeepMind, to predict the effect of a variant. And of course, these are predictive tools, only time will say how to use it for future. And next, what I would tell is, this is the power of language model, like why ChatGPT has been so powerful. This one, using all the text which was there anywhere in the world, a machine learning model has been trained. And what it required is a field called computational linguistics. You have to put things like the king minus man plus woman is queen. What it is telling you is, you are using the text, making it into vectors, which is called embeddings or something. So now you build context into it. So like the king is used in certain context, queen is used in certain context. If you train the literature, you are able to bring that. So a machine learning is able to capture the context, and using that it is able to do predict. And same thing is happening, and there are tools like Grover or things like has been developed, which can use genome as a language, and using that we can find new patterns or something. So in addition to getting a sequence, we should embrace all these newer technology and explore whether they are really powerful, can they solve societal problems. So I'll stop here.
Thank you very much, Dr. Debashish. I think the panel has given enough of information for discussion. So now I invite all of you to actively participate and discuss with the panel. Mike. Okay, so, okay, yes. So there is a mic. If anybody want to ask question, please raise your hand and we'll reach the mic. Yes, Professor Narahari, then next. Yeah, please. Here, first row. So while it's coming, I thought I'll ask one question. Okay, please.
Good morning. This question is for Professor Ramesh Sonti. You were talking about plant genomics. Turns out that soil, temperature, rainfall play a very important role in the growth of plants. How does plant genomics actually incorporate all these factors while doing all its studies?
Thank you. Thank you for the question, Professor Shastri. I was referring to the genomic selection tool. Okay. The challenge as we move forward with climate change is that in a single season, you're going to have a crop that is exposed to flooding, drought, as well as high temperature. So earlier, if you could make a variety that is tolerant to drought, that is okay. Then because some areas had only drought, some areas had only, heat stress. So different, or submergence. Now we're actually, with climate change, things have become very complicated. So you will have to have crops that are tolerant to multiple stresses. One way is to screen a large population. So traditionally, the way breeders go about it is that they have a large set of germplasm, they screen them and find out which ones have the traits of interest, and then from that they move them into the commercial varieties. So now what people are doing, because these traits are so complex to phenotype, they're essentially and very expensive, they are now using machine learning methods for this. So you have a large population, but you take a subset of it, and that subset you now do the extensive phenotyping. And you get genotype data for the whole population. And based on that, now you use machine learning methods trained on the test population, on the training set. Now you elaborate it, extend it to the whole population. Pick up the ones that appear to have the traits of interest. And once you have identified a variety, then through genetic methods, coupled with genomics and bioinformatics, it's not very difficult to map the traits of interest. And once you mapped it and you have a DNA marker next to it, you can move the trait into any other variety. So that, I would say, is the way things are moving right now.
So just one general question to all the panelists. This is really a very, I think, a wishful project, that's Genome India. I just want to ask that, is this particular platform working in something in relation to that One Health approach, like when we talk of mutations and all that. So, what are the triggers? Like we very casually say that, okay, there are rising temperatures, the crop failures, the drug resistance problem in the various, susceptibility of the various crops to various pests and all that. And so there has to be some kind of, like we also we are talking of Genome India, we also talk of the epigenetics, where when we are taking of the environmental, triggers, the things are falling back to normal level also. And has there been any effort at the therapeutic level also? There's a lot of work on CRISPR technology, gene editing. There's a lot of work. We are talking of, so many inherited disorders and all that. So is there any work, that has been undertaken by this platform?
Right. Go ahead then. Yes, particularly for treatment aspect, there are a couple of things actually going on. One is gene editing for sickle cell. IGIB, CCMB, and a few other organizations are part of it. Then for the hematological disease, CMC is doing gene therapy. So these I know. If anything else is there, Prostelma can. Yes, there's one. Yeah, after this.
I am Director, Sher Kashmir Institute of Medical Science. When Genome India was being constituted, I was the only clinician that time, so I had the data which we collected. Do you think our phenotyping of the individual is as good as second thing I want Shinjini particularly and all panelists in US one screening programs are defined, well defined. For example, in US there are in each state has 60 disorders mandatorily to be tested. How far we are in our country and having so much heterogeneity in the population, can Genome India be a guide towards screening program?
Okay, thank you for that. So maybe I can answer for the first question and leave the second question for Prostelma and Dr. Shinjini. So when we collected the samples from 20,000 individuals, Yeah, before that, when you talk about normal individuals, I think none of us are normal. We all carry thousands of mutations, right? Sometimes it triggers and causes the disease, sometimes not, because of many other reasons. So these are the self-reported normal individuals. But still, after collecting, they might have had some disease. So to have some kind of homogeneous population, so we have just took that approach, right? So that's what I wanted to say for the first question. But for the second question.
Okay, I will address your question on newborn screening. I made a comment in my presentation that in India we do not have mandatory newborn screening. And the basic reason for that is we do not have the epidemiological data. There are no prospective studies which have been done anywhere in the country which will actually say that this particular state, these are the kind of conditions which are prevalent. Most of the times, as I said, the NICU babies are coming and that's how the data has been collected. So the answer to your question is, unless we do a state-wise epidemiological data generation for all newborn screening tests which are available, as of now 80 to 100 are available with just 1 drop of blood, it's only then you're going to be able to pick up. And then the question of, bias won't come because every consecutive newborn is taken in the study and taken forward. So that's really the reason why newborn screening is not there. And our data showed that out of the 200,000 babies, we got about 15 different conditions. And on an average, our study saved 1 in 200 babies. And we've been trying to say that this is very important to push it. So that's why the UMID program, we are hoping that will really educate doctors to kind of take it up in a more proactive way, because it is an example of proactive medicine rather than reactive medicine. You're already looking for identifying based on screening and then already giving the pre-symptomatic treatment so that at least 15% or more of these conditions can actually be treated and they could just live normal lives.
I'll just add, I think what Dr. Thelma is saying, there's a lot of value in that, but there are huge challenges and caveats for newborn screening. If Madhulika, would you like to make a comment, because she's been a backbone of newborn screening on this? Yeah. Madhulika is head of Department Pediatrics at All India Institute of Medical Sciences and has started the genetics department at AIIMS.
Thank you very much, Shinjini. So efforts are on. As Shinjini said, in our country we do not have a national newborn screening program. But I must say that there are states which are offering newborn screening. Like you have a program called NEEV in Delhi, where, apart from congenital hypothyroidism and congenital adrenal hyperplasia and G6PD, 5 more disorders are being screened. As Elma was referring to, the with the DST program in which 200,000 data apart from the 5 disorders. Then there are individual have it for born babies only. There are programs for newborn screening but where that what they are trying to do, and I think that's a better approach in our country, that they are doing a comprehensive screening. So they're not sort of only focusing on DBS based tests where they do metabolic screening, but at the same time they would also do other screening like externally visible and retinopathy of prematurity, combining a universal comprehensive newborn screening approach. As far as the genomic newborn screening is concerned, which we call as GNBS, is actually not a recommendation as of now, because there are many issues with that. One is that if you, as has been said in the morning, that if you do a genomic screening, you will get multiple variants, right? And what to report, what not to report, what is action. If you find some things like predisposition, say, to cancers, what do you do in the newborn period? So there are I mean, there should be guidelines, and there are guidelines in process where they would say, okay, these are the disorders which you should report if you are doing genomic screening, for which immediate action is required and maybe some treatment is possible. So that is evolving. We cannot jump onto genomic newborn screening, but for our country, I think many efforts have been made. The ICMR program was the first program, multi-center program, where only 2 disorders were screened, but now it is picking up. Even Kerala has a program. Many other states have. Thank you.
Thank you, ma'am. In the interest of time, okay, we can have last quick question and then probably we'll wrap up the session. Please introduce yourself and ask the question.
This is Dr. Mukesh Kumar Meena from NIPGR. I'm plant biologist, but I'm curious, like 83 population group is included in this genome sequencing project.
99. Huh? 99.
99. 83 was written in that leaflet. So okay. I was just curious whether this native population of Andaman and Nicobar was included in that, because it's a distinct group which is not touched by like other world people. So it might give some interesting information actually. So is it included, this native population of
islanders are not included in here.
Actually, in Genome India project, we have covered populations all over the country. And as per the policy, I think maybe details will not be given for the populations. And this is as per the population, as per the policy of the government. Okay.
But those set of peoples are not included.
That we will not be saying. The population, about population description. But definitely population is covered all over the country.
Dr. Shuchita, we can say which is not included. So definitely it's not included. So we will not say which is included.
Okay, in the interest of time, we move on, we wrap up this session. Before we wrap up this session, I request Dr. Jyoti Malik Logani, Scientist F DBT, to kindly felicitate the panelists. I request Professor Tangaraj to please come in the center. Yeah, ma'am, head this way. Ma'am, aapko bola hai. Aage se do, aage se do. You expect us to Professor Telema. Dr. Shinjini from THSTI. They're all same. Sorry. नीचे से दो, नीचे से. Ma'am, we can swap. No, this is my fault. Professor Santi. Dr. Debashish Mohanty, Director NI. Thank you, ma'am. Let me announce tea, then you can have a group photograph. So we break for tea here, and we'll be back in 15 minutes. Thank you. Tea is arranged outside in the atrium. Panelists are requested for a group photograph.
Are you happy?
Yeah, ma'am. Very happy.
Yes?
Very happy. It's going according to schedule. Please for a photograph.
सिलेक्ट करेंगे ना उनको सुनाना पड़ेगा, तो पता चलेगा, नहीं तो हमें हम वो बड़े वाले सिस्टम में बैठे हैं ना।
ये नहीं इससे लगे रहने दीजिए, उसमें होगा ना, स्लॉट है नहीं है, स्लॉट नहीं है तो
उल्टा है सर।
कुछ पता नहीं चलता है भाई।
उधर लगा के आ रही है।
ब्लैक कहाँ हो रहा है?
जैसे कि अभी हो रहा था ना वाइट सा और लाइट हो रहा था।
बताओ
अब ठीक है। तो आप
पहले भी यही था, कुछ छिड़क हो रही है यार।
वो बोल रहा था
भी चेंज नहीं
3 बार कंपनी।
नहीं कितने से हाउ मेनी प्रेजेंटेशन आर देयर इन दिस? एक तो इनकी
टू विल यूज स्लाइड्स।
क्वेश्चन वाइज भी किया हुआ है, तो क्वेश्चन वाइज अलग-अलग बुलाएंगे, दो पर बुलाएंगे।
उसी
तो एक ही पीपीटी में
एक ही मिनट में
नहीं वो
एक
अलग
करके
वाला
वो
एक
यहाँ से उसको बता देंगे कभी क्या होगा। तो आप अपनी स्लाइड्स भी उसी में ऐड कर दीजिए, जो पेनल्टी की स्लाइड्स एथिक्स के।
लेकिन वो ऑर्डर वगैरह वो नहीं पता मुझे,
हेलो
पूरा हिल जाएगा।
Slides are in response to the first question or second? So I think Shantanu will be one and yours will be two. We have just 2 presentations.
और फिर जो बाद में सेकंड क्वेश्चन आप मुझे वापस
सेकंड क्वेश्चन में ही होगा ना आपका?
नहीं फिर 2-4 क्वेश्चन बाद आप वापस मुझे दूसरे क्वेश्चन डाल दो।
You want to add one more question?
No, I am not adding one more question. तो आपने तो ये भेजा है।
तो टंगरा सर ने किया ना उसमें। हाँ उसमें है। उसमें ये वैसे ये इस पे खुला, इस पे खुला हुआ है बस, ठीक है। पढ़ लेता हूँ पहले, फिर कॉन्फिडेंस से बोल दिया था एक बार पढ़ा हुआ पहले तो।
उसके बाद ये आपसे एक क्वेश्चन डाउन द लाइन आता है, तो इसी के बाद रहता है तो मेरा वो
ग्लोबल एथिक्स ऑफ डाटा शेयर। यार मेरे क्वेश्चंस क्या हैं इसमें? मैंने क्वेश्चंस ही नहीं पढ़े अपने।
ये वो मैंने पीपीटी डाउनलोड किया यहाँ पे तो वो चेंज हो रहा है, मेरे को पीडीएफ डाउनलोड कर दिया। एक बार चेक कर लेते हैं सर।
भाई अभिषेक जी इधर स्लाइड चेंजर लाना वहाँ से, स्लाइड चेंजर रखा है आगे रखा है। आगे करना
हो रहा है ना,
और आगे करो, इधर इधर चल रहा है, आगे करो, आगे करो, आगे
कर दिया
कितने पेज?
हाँ ये बस ये लास्ट पेज ही होगा।
अच्छा लास्ट, बैक करना, बैक करना, बैक 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 बैक। ठीक है, तो ये स्टार्टिंग से चलाना ना फिर आपको।
ये वाला ये मैं जब आऊँगा तो ये पीडीएफ फाइल चलेगी स्टार्टिंग
पार्ट्स में होगा। फर्स्ट पार्ट में फर्स्ट
ये पीपीटी है ये चलेगी।
तो ये ठीक है, एक ही पीपीटी में है ना सब पैनलिस्ट का?
दो में
ओके,
सर का जो
एक
है वो
पीडीएफ
आप ठीक है, पैनलिस्ट 2 है ना, अभी
2 नंबर पे शुरू हो जाएगा। जब वो मुझे बुलाते तो वो पीडीएफ फाइल खोल जाता है।
आपका नाम है सर?
मेरा नाम स्वप्निल राणे है।
स्वप्निल राणे,
ये इस ऑर्डर में जाएगा ऐसी गारंटी नहीं है।
इस ऑर्डर में नहीं जाएगा, मैं मॉडरेट करूँगा।
ओके,
तो फर्स्ट क्वेश्चन जो होगा वो उसमें
तो फर्स्ट ऑर्डर में यही आ रहा है।
फर्स्ट
मैं आपको दिख गया
जब इनका नाम बोलेंगे
तो आपको ये वाला
जब हम इनका नाम बोलेंगे,
अच्छा पीडीएफ चलाना आपके केस में?
मेरे केस में पीडीएफ चलाना है। ठीक है, ये जब बात
दो बार नहीं, मेरे कोई स्लाइड्स नहीं, 2 ही स्लाइड्स हैं। आई डोंट
ये रिस्पॉन्सिबिलिटी है किसकी?
या रिस्पॉन्सिबिलिटी शांतनु,
ये डॉक्टर शांतनु सेनगुप्ता का।
तो जब फर्स्ट आप ही का चलेगा, जब मैं बोलूँगा स्लाइड स्टार्ट करने के लिए तो सबका स्लाइड चलेगा।
तो आपका स्लाइड कौन सा चलाना है सर?
1 to 4 चलाना है।
पीडीएफ आपका ये खोल गया
है। स्टार्टिंग की कुछ स्लाइड्स हैं जो मैं चला लूँगा वहाँ से,
आप आगे आगे
कर लूँगा और मैं फिर रुक जाऊँगा।
ठीक है, उसके बाद पीपीटी चलेगा।
उसके बाद फिर
दूसरे क्वेश्चन में मैं बोलूँगा पीपीटी स्टार्ट कर दीजिए।
डॉक्टर शांतनु आएंगे तो पीपीटी स्टार्ट कर देंगे।
नहीं, मैं बोलूँगा क्योंकि उनका फर्स्ट क्वेश्चन में उनका पीपीटी नहीं है।
ठीक है, ठीक है, तो डॉक्टर शांतनु में पीपीटी चलाना, आपका पीडीएफ चलाना।
और मैं बोल दूँगा आपको पीडीएफ से।
नंदन को बोलना स्मार्ट लड़का मैंने अपना लड़का बुलाया है मेरे से दो लड़के मैं करा दूंगा लेकिन वो स्मार्ट लड़का था कि बोले सर तो बस मैं करा दूंगा कैपेसिटी बस करा दूंगा वो करा दूंगा बस ठीक है मैं करा दूंगा और सर एक काम करो जो छोटी हाइट वाला है ना यहाँ पे नीचे डेस्क पड़ा हुआ है ये नीचे पड़ा हुआ है इधर ऐसे करा छोटी है कोई हाथ में खड़ा हो तो ही कर सकता है वॉलंटियर कोई है
पे पीपीटी चलेगी, फोल्डर खुला होगा। उनको बुला लो सर, हाँ।
लाइव चल रहा था एक इसका वो दे दूंगा मेरे को लैपटॉप का कुछ होगा ना जी टैग वगैरह कुछ भी नहीं है बैकअप में डाल देना ना फालतू टाइम पे
Good morning, welcome back. I request all to take your seats so that we can start with the next session. Good morning all. So the first session was dedicated to understanding the significance of genomic research, and we learned about how important it is to the understanding of genomic research for predictive medicine, for developing new climate resilient and improved varieties of crops, and also how it's important to generate new tools for developing understanding these genomic data. So now the next session is focused on a discussion on global ethics of data sharing. Historically, the concept of biological data sharing can be traced back to the early 20th century when scientists began to recognize the importance of sharing research findings and experimental data. The establishment of scientific journals and the advent of digital technologies have further facilitated the exchange of information, paving the way for more collaborative and interdisciplinary research. Biological data sharing is a critical enabler of scientific progress and innovation in the life sciences. Our next session dwells into the responsible data sharing, especially in wake of the data generated from the public funded research, and this research should be judicially used. So for the next session, I invite Professor Shandar Ahmed from School of Computational Biology and Integrative Sciences, GNU. Sir, please. Dr. Shantanu Sengupta from CSI IGIB. Professor Deepak Nair from RCB Faridabad. Dr. Swapnil Rane from TMC Artrek, Mumbai. And Dr. Richi Mahajan from DBT. Dr. Shanda will moderate the session. Thank you, sir.
Thank you. So in the morning you were hearing about the importance of data and how genomics data can be so useful and important. DBT has been working and together with so many scientists, and we heard that the efforts of PRIDE guidelines for sharing the data and the protocols which were specific for the formats to be used for sharing this data. We have in our panel some prominent speakers, Dr. Shantanu Chaudhary, Shantanu Sengupta. I'll introduce them briefly. Yeah, before I introduce, I would just say that it was not long ago, until long ago, that data sharing was really there was a lot of resistance towards sharing the data. And in 2017, there was an article in New England Journal of Medicine about data sharing. I was discussing this in CDAC yesterday. And in that article, it was mentioned that the data scientists are data parasites. And the primary reason that they are data parasites are they are not generating the data, they are analyzing the data, and sometimes they are proving the original hypothesis of the data generator wrong. So in that sense, they are very much similar to parasites. But the debate started, and next year there was another article in PLOS Biology, which says that actually all biology is computational biology, and there can be no integration of science without having data integration, data sharing. So we have come a long way in such a short time, and data sharing has become very important. And it has been now recognized there are so many important reasons that data must be shared. First of them being the cost effectiveness and duplication of the scientific activity. And then the data became even more important with the advent of artificial intelligence, AI, because most of the AI and ML models today are based on data, and therefore the responsible and appropriate use of data and making the data available for those who can develop those models has become important. Many international efforts in that direction have been happening. Elixir in Europe, Open Science Foundation is working on how to share the data, and the focus, different focus topics have been there. For instance, focus is also reproducibility of research. Focus is also on the ethical biases, the data biases. Therefore, any scientific outcomes may be more appropriate for one particular population group. So from that perspective, and we heard about genomics data in the morning, but this is not just about genomics data. Any types of biological data has these issues, concerns. On the positive side, that we want to have reproducibility, we want to have the sharing and the model development, we want to have open science. We also have the concerns to the counterpoint of the perspective, which is about privacy and the security concerns. Whether the data, if we develop some data which is sensitive, whether it should be shared and how can we responsibly share, who are the people with whom we should share the data, and then the question of how to share the data. So first is whether to share the data or not, what kind of data to share, what are the pros and cons for sharing the data. Then the question is how to share the data, how to write our data in a way that others can use it, what are the minimum standards of the formats, what are the minimum information that should be present in the data so that it becomes usable and it becomes kind of applied in the models. So these are the methods. Earlier, this kind of approach was taken by journals. Several journals set standards about asking researchers to deposit their data into respective repositories, PDB, for instance. There came the Array Express or Gene Expression Omnibus. The minimum standards were developed specific to disciplines. Consortia came forward, minimum standards for writing the gene expression data, etc., and NGS also. So several journals and consortia has set up standards to how to write this data so that it becomes shareable and it becomes reproducible. The studies become reproducible from that. Then the questions in this regards also come whether the data sharing, data deposition, should this be voluntary and should it be mandatory, and what is the role of the funding agencies in this? And these are the issues I think we all have been dealing with, and these all concern us. So the question comes in the technicality of the data sets, the need for the sharing of data sets, but also the implementation and the legal and the ethical aspects come into picture because of this. So I have in my panel Dr. Shantanu Sen-Gupta, who will be talking about the ethical necessities and ways of data sharing. So I invite him to speak about it.
Good afternoon, everybody. Let me thank DBT for inviting me here today. So, as Dr. Shandar Ahmed was mentioning, the concept of data sharing is not old. It is just fairly recently, this debate has started. And more researchers are convinced that data sharing is important. For me, data sharing is important for my science also. So the question of whether data sharing is important or not, I think that debate has been rested. What we should now think about is how to share the data. So today, I'll tell you, when I do a metabolomics or a proteomics experiment or a genomics experiment, the journals ask me to just share the data or store the data in some database. And I just take the data and store it from there. Is it sharing? No, that is actually not the sharing we are talking about. if you have to share data, it has to start from much before, especially for human samples. If you, and I know there is no question of if, we should all plan to share data, and this should start from very beginning, from writing your project. The funding agency should have one column now, how the person is going to share the data, what are the protocols that are involved, that he is going to make sure that the data is being shared. After that, then comes collection of samples. You know, the basic premise is that the owner of a information, of one individual's information, is that individual. So you cannot take something from a person without informing him, right? That's called cheating. That you will be in jail. Similarly, in case of data also, because he is owner of that data, you cannot take the data or share the data unless you have taken permission from him. And that's where informed consent comes into play. And often I have seen in many studies in the country even today, they just go to a patient, say that isme aap sign kar dijiye. And the participant does not have even 5 minutes to read. And also often you go to population which doesn't understand English or doesn't understand the medical terms, and you put the medical jargons there and you ask him to sign, he signs. So we need to start from there. We need to have informed consent prepared in a manner which is actually understandable to the community that you're going. If a majority of the community or people there are not literate, you need to explain to them that you're taking their information, and there comes the second point, that is transparency. You need to tell them why are you taking the data. I can give you an example. In 2022 end, CSR launched the Genome India project, and this was one of the major things because we were collecting samples from across the country. So wherever we started collecting the samples, we followed two ethical principles. One is we went to all the places and first gave an awareness lecture that we are taking samples, this is what we are going to do with your samples. Then we had created pamphlets in different Indian languages. You know, it is very easy now, you just go to Google and it will translate and give it to you, and then just ask a person, native language person, to translate it. So we had created posters of that, and we had pasted it. And then when the day of sample collection, prior to that, in the portal where the person was registering, we had pasted the informed consent so that they can read it beforehand, before coming for sample collection. That is important, and I think that kind of guidelines we should all follow. The next is privacy, and I think this is one of the biggest and most challenging thing, and we should all start following. I have looked at several studies, both national and international, which do not follow the prescribed privacy protocol. For example, you collect a sample from a person, the name of the person is written here, and everything is written, and the PI, then based on that, he is doing a genomic experiment, so he writes the name of the person and gives the genomic data, and that circulates among students, and this, he will say that I have not shared it beyond, but he has shared it with his students, he has shared it with himself. So what needs to be done is that you will have different numbers. The moment the person registers, his name is given, he is given a registration number, and from that time onwards, the registration number is dealing to the personal information. Now from the registration number, when he is coming to give the sample, computer generates a second number, which is a lab or test ID. Once it goes for various assays, various genomics or proteomics or metabolomics assay, a third number is given. So under no circumstances the person who is doing the assay will ever be able to understand whose sample it is. If I am taking my sample, unless I give the number through the computer, I should also not be able to know what my data is. And then the data, when it goes back, whatever data goes back to the participant who has given the sample, it goes through the computation steps, test ID to lab ID to registration ID, and it goes to his portal. But this kind of ethical, so de-identification of sample is very important. And there are certain protocols for de-identification. In some cases, you might want to make it anonymous. In some cases, de-identification is enough. We need to understand the difference between the two. And, time has come. Literature is there. PIs who start a project, my sincere request will be to understand this. And I have seen, believe me, I have seen NIH studies which are not following that. And I'm glad to inform you, one NIH project has now followed our protocol and they are now implementing there. So that is very important. Privacy of the person is important. Next come intention. Why are you wanting to do this study? Your intention should be good. Your intention should not be malified in terms of you should not try to collect the information for your personal benefit. Sell the information and get money. That is one of the biggest challenges, not in genomics field, but in every other field we see. We don't know from where our phone numbers have gone, and people from this bank, that bank starts calling, and we get, so we need to be very sure what is our intention. And even if intentions are good, sometimes we cannot share the data because the outcome may not be that desirous. For example, especially this is for genomics, you get a certain mutation for a particular community. You just start sharing the data with the name of the community gets, branded that they have this mutation, and that is not ethically correct. There are many more things, but these are the basic 4 or 5 principles that I feel is important before you can start a study. With that, I'll hand over it to Shandar.
Thank you very much. So it was a nice motivation to start with the discussing the question of ethics. And as I said earlier, ethics and law sometimes overlap, and we sometimes cannot distinguish between what is legally valid versus ethically valid. So in that regards, a lot of data, medical data that Tata Memorial Center is dealing with. Here, Dr. Swapnil Rane will talk about the issue of the ethics as against law or as overlapping with law.
Thank you for inviting me here to this meeting. Can we have the PDF displayed, please? Right. So I started my medical career dealing with a lot of these issues, and we do a lot of research as well as do diagnostic practice and clinical practice together. And we often are faced with these kind of questions very regularly. What is ethically correct versus what is legally correct, and ethics and laws often they do overlap, luckily most of the time, but there are situations where ethics and law may be opposing to each other. I'm just going to try and highlight some of these areas where these things happen often. This is not an exhaustive list. This is just a small list of the most relevant things that are relevant for genomics as well as for research in general.
I think there's some technical glitch there.
All right. So I'll just talk about it. So ethical principles are guiding principles. They are not the rule of law. They are very generic. They are often very context specific. So the ethics can change depending on the population, depending on the country, the society that they are in.
a lot of these things are very fluid, and that's why they are interpreted in context. So the same activity may be ethically correct in one situation while not be ethically correct in another situation. So this is very context specific, and therefore you usually have external ethics committees or institutional ethics committees who are not involved in the study at all, looking at these aspects. So they look at all of these aspects and try to guide the principal investigator or the investigators as to whether the study that is proposed is ethically viable, correct or not. And if not, then they suggest what to do or what is allowed and what is not allowed as per the ethics committee. Laws, on the other hand, are very rigid. Once it is written on black and white, you have to follow it. There is no scope of interpretation as long as there is no exception written in it. So if an exception is written in the law, then it is allowed. So for example, sharing of genomic data is a very significant point, and not just about genomic data, but any personal data. So what is personal data? Any data that can, by itself or together with other data points, lead to identification of an individual is a personal data. Any data that cannot lead to the patient or a person is non-personal data. So sometimes you have patient information. For research purposes, you don't really need every patient data. Then you can have aggregated values. So you can have mean of the population, median of the population being used instead of actual data. You can have geographic distributions of the data of the patient's disease profile, etc., being provided, and then that is not personal data. But there are these scenarios. For example, by law, most of the laws in almost every country require explicit consent for sharing of data. Can you please not move the slide, please? Okay. So many times we have taken consent, we have done a particular study. Now that study I want to put in a repository. In that repository, any third party can also access. They can use it for another study. So the initial explicit consent was given for study A, but the explicit consent for study B was not given. So by law, explicit consent is required. There are few exceptions in some of the laws. For example, the PDP law has a certain exception for processing personal data for functions of the state. Now, COVID is one of the situations where this was probably applied. But this thing is, as a general rule, whether it is HIPAA, whether it is GDPR, whether it is Indian laws, they require explicit data for sharing of personal data. Now, this extends even to family members. So, for example, a particular mutation may be relevant for the family because the family needs to be screened and family needs to have certain tests to be done. Now, we cannot do this without actually telling the patient that we are going to tell the family. You cannot do that by law. You have to tell the patient first that yes, we would want to share. If you provide consent, we will do that process. Historically, we might have collected a lot of data. Laws keep changing, rules keep changing, and certain data might be collected which was collected under less stringent laws. Or perhaps there were no laws related to it. Now, as you go ahead, you have laws that have come up, and they apply not just to prospective data collection, but they also apply to all the retrospective data that was collected till date. So these issues always keep on coming up. Many times data is shared across boundaries. In India and in China and many other countries, there is now emphasis on localization of data in the country. So that is one of the important factors. But when data is shared, the benefit to the researcher is significant, but the benefit to the patient or the population from which the data was derived is not very evident. Especially, the benefit is often not there, or it might come back to the population maybe 10 years or 20 years down the line. But the patients who have contributed the data do not really benefit from the studies. So this is one of the ethical and legal issues that are always there, and there are no clear-cut laws regarding all of this. Benefiting from the monetary benefit that also arises from research. So many investigators will go on to patent their publication, their work, and they will have companies built around it and have monetary benefits coming out of it. Now, it is not very clear as to how this is to be dealt with and how certain benefits can go back to the population that actually provided the data. So there are no laws, there is no clear-cut ethical guidelines as well as to how this will happen. There is in general a principle that yes, the population should benefit, but it is not clear how to do this. So there are certain protocols being set up for international data sharing, where like the Nagoya protocol, where this kind of feedback or the benefit being sent back to the population from which the data is collected. Certain thoughts and certain laws have, certain guidelines have been set in it, but there are no clear-cut laws and there are no treaties regarding it. So I already mentioned what is personal data. Every law defines it in different ways. In general, anything that you can personally identify the patient is personalized data. Is anonymization enough to say that a data is no longer personal? It depends on the degree of anonymization and the risk of re-identification. So if it is pseudo-anonymized, but somewhere in the backend database or somewhere else, you are maintaining a link from the anonymized ID to the original ID, then it is not non-personal data, it is still personal data. So this is my thoughts on the issues that are there between ethics and laws. And there are no clear, this will keep evolving. And as we evolve, there will be more clarity, hopefully, regarding these issues. Thank you.
Thank you. So as we understand, no society can work simply based on the law. Ethics beyond law are important, and thank you for highlighting some of the important aspects of ethics law and how sometimes something that is legal can still be unethical and vice versa. So now we motivated ourselves to think about the question of ethics and how it is different from law, etc. So, but now even with the best intention of data sharing and the data management perspective, we cannot do it without technical infrastructural support. So what are the technical concerns and how we manage large amounts of data when we are dealing with population data and genomics and any kinds of imaging and different types of data? What are the challenges that we face? So we have IBDC here representing Dr. Deepak Nair, and he will share with us his experiences from IBDC. What are the technical challenges and issues that are to be addressed in dealing with data sharing?
Good afternoon, everybody. My name is Deepak Nair, and I am the current head of the Indian Biological Data Center. So the Indian Biological Data Center, IBDC for short, is the digital repository which is funded by DBT, which will archive all the life science data generated in India. And currently we have 5 petabyte of space available. The center is located on the campus of the Regional Center for Biotechnology in Faridabad, and we have 5 petabyte of space at Faridabad and 1 petabyte of space at our disaster recovery site in NIC Bhubaneswar. And we have set up portals to receive data, and now we have 7 portals active, where people can deposit nucleotide, proteomics, imaging, 3-dimensional structural, and metabolomics, as well as crop phenome data. And this database has grown over the past 3 years now, and we are ready for a round of expansion, and we hope to increase the capacity by 5 petabyte. So there have been multiple challenges in setting up the data center, and couple of the major ones is that there has to be a culture of data sharing, which has to be inculcated.
Slides off, please, because this may be misleading for the
Yeah, I don't have any slides.
Because live stream, so we should not have slides.
So I would request all of you who have data to kindly deposit data in IBDC. And I am a structural biologist, and all structural biologists will tell you that for about 50 years we have been depositing data in the protein data bank, and the benefits of that are immense, and the effect that they have had on all sorts of applications, fundamental as well as applied science, has been tremendous. And you can see that how nowadays these artificial intelligence programs have benefited from the data available submitted by structural biologists. So the case is, meaning if you study the history of structural biology, you can get an immediate idea about how beneficial it is to share data. The other is more technical, is internet connectivity is always a major issue, and we have to struggle to find ways and means, although we are close to multiple redundant solutions now, to ensure that everybody has ready access to the databases. And the data is stored at IBDC, and IBDC will help all the individuals who want to share their data, who want to archive their data at IBDC. There will be people, there are teams which will help you share the data. And IBDC is like a service provider for the DBT. So DBT, with the help of relevant experts, will make the policy, and IBDC will execute it. And that is what is going to happen for all the human genome data also. And we are mandated, as from an ethical point of view, we are mandated that we cannot accept any data that can be used to identify the individual. And so the depositor has to ensure that all the ethical issues are taken care of. So this is a brief overview of the IBDC and its activities. And again, I'll request everybody who are watching online also to deposit your data in IBDC, and you can see how it will have a lot of benefits to your own research as well as the overall the level of research that is conducted in India. Thank you.
Thank you, Dr. Nair, for sharing with us the technical issues in data sharing. And now, worldwide data sharing has faced hurdles because people who actually generate the data, they don't want to share it sometimes. Sometimes they don't want to share it because they think they can still churn out some more interesting intellectual property out of it. Sometimes they do not want to share it because they are not confident if the data will be proved wrong or their studies will be questioned afterwards. Whatever is the reason. So the practice of sharing the data and the enforcement and trying funding agencies and stakeholders, those who have interest in making this data public, are faced with the policies. So we have here Dr. Richi Mahajan, who will talk about DBT's effort in trying to convince the scientists to deposit the data and how they're enabling this process.
Thank you, Professor Shandar. Can I, before I start, can I ask technical team what's the issue going on? Why is the feed not on? The technical team. Why is the feed not on? Is it going live also? Because we have a keynote session after this. This would be a problem if this continues such way. Anyways, thank you, Professor Shandar. So as you know, government is spending huge amount of money on public research, and huge amount of data is being generated. According to an estimate, approximately every year 40 exabytes of data is being generated. And this has to be shared, because if we keep duplicating the efforts, it's going to have a lot of it's going to incur a lot on public exchequer. So the data sharing has to be there. And for this, government came out with a policy of NDSAP, National Data Sharing Policy, back in 2012. And the main features of the policy was maximizing the use of data, avoiding the duplication, maximizing integration, ownership information, better decision making, and equal data for all. So the key principle was actually the timely release of data so as to accrue the maximum benefit. Now, keeping in view, this policy was in general applicable to all kind of data. That was not specific to biological data. Keeping in view, DBT started its effort back in 2016 by we were actually trying to come up with a policy, but then we were told that, there exists a policy, so we need to pursue with the guidelines under the ambit of the NDSAP 2012, which was notified by DST. In 2021, after an extensive interministerial consultation, expert consultation, stakeholders consultation, the policy was there, the guideline draft was there on public domain for a lot of time, and after probably humongous rounds of these deliberations, and all credit goes to Dr. Ninave and team, these guidelines were released back in July 2021. So they were called Biotech Pride Guidelines, or what we call is the promotion of, So these were called the Biotech Pride Guidelines, or what we call the promotion of research and innovation through data exchange. Now, these guidelines broadly tells us about the data, how the data has to be shared, what data has to be shared, what are the kinds of data, and they list around 10 to 12 kinds of data, but then they do not limit itself. I won't dwell much into it because there's this presentation by Professor Sahu in keynote session on this whole thing, but I'll just mention the efforts of funding agency and then come how the funding agency is actually trying to implement the data sharing and the efforts made by the funding agency to implement the data sharing. So coming back to the guidelines, these guidelines basically talk about the types of data but does not limit itself. It says all kind of biological data. One thing I again like to highlight about these guidelines is that these guidelines do not deal with the generation of data per se. They mainly focus on the sharing of data which is already generated. Okay, and these guidelines have been prepared in agreement to all the national and international. There has been an extensive consultation, and these guidelines have been prepared in harmonization with all the national and international these agreements. But however, in case of misalignment, the national policies and laws will supersede. That has been very clearly mentioned. Now, there existed everything, but then, and these guidelines only actually, kind of in its para 5 mentions about establishment of data center or probably sharing of data, the nuances of sharing of data. So this data sharing was mentioned in that, and that is how IBDC came into picture. Indian Biological Data Center came into picture. So then after the establishment of Indian Biological Data Center, still the question remained was, what are the protocols to be? And this pilot study was all carried out for actually establishing the SOPs and protocols for sharing of data. Now the question was how to go about the sharing of data. Then as per the PRIDE guidelines only, the framework states that there would be a data management committee which will actually report to an expert advisory committee. And I'm not presenting my slides because everything, the feed protocols is, these PRIDE guidelines are very well available. You just type Biotech PRIDE guidelines, it will be the first tab which opens. So these guidelines actually state about that there would be a data management group followed by, which will report to, which will apprise expert advisory committee. And this expert advisory committee will then apprise to interministerial committee in case of any misalignments. And this this national steering committee or the interministerial committee has, MHA as the observer. Many of you, I would congratulate all the members of data management group. We had extensive consultation. Professor Shandhar led the show where he drafted the zero draft, and along with Professor Shantanu and Dr. Dinesh Gupta, who's amongst the audience. But it was equally contributed by all the data management group people. We had series of around 13 to 14, I've been pinging them up every time of the day. And we had series of 13 to 14 discussion meetings. Dr. Ninave actually, kind of gave all her time for this whole effort. And finally, we could come up with a draft, which was then again extensively discussed in expert advisory committee, and then, kind of recommended by Interministerial National Steering Committee for implementation. What we call now is the feed protocols, or framework for exchange of data. Now, these feed protocols basically tells you they actually strikes out the main responsibilities of the data submitter. So basically, onus of data sharing falls on 4 people: data generator, data submitter, this funding agency, and obviously the data user. So there is a striking, there's a balance which has been kept, and all the duties have been assigned in these feed protocols to all the stakeholders for responsible sharing. Coming to how funding agency then try and implement this. We have actually all the sanction orders, what we are issuing. We cannot be sticking around everybody, but still we have in all the sanction orders, what we are issuing, we have made it mandatory for all the public funded research that all the data, all the primary data which is being generated has to be submitted to Indian Biological Data Center as per but we cannot bite everything, right? We have few portals up as on date. I think 8 or 9 of them. Professor Nair would be able to tell me more, but we have 8 to 9 portals which are right now currently working. So as on date, we are accepting those data, and we are coming up with, again, specific more portals depending upon the availability, technical availability. So we have made sure, and there have been a lot of talks of, as Professor Shantanu said, Dr. Shantanu said, mentioned about that there should be a plan before, the data, project is submitted. So feed protocols does talk about that as well. So I think that is where I rest my case, that we're trying our best for data sharing, and I request you all to please go through these guidelines, to go through the IBDC website, so as you can actually get into the details of the data sharing. And believe me, I mean, sharing your data would actually add more value to it rather than, kind of losing your kind of research. Thank you.
So thank you, Dr. Ritchie, for your overview, and I congratulate DBT again for spearheading the initiative for data sharing. And this could have been done by many other agencies, but DBT has come forward and taken an initiative which is really very welcome and very timely. And thank you also for exchanging the congratulatory note amongst each other, and also not missing an opportunity to convince you people again to share your data. And so I'll take a second round of quick questions and touch upon a few more aspects of data sharing. I request panelists to restrict to 3 to 4 minutes in this round. So what we have, the topics that we would like to cover is the AI, because data is very closely connected to AI. And then we will talk about the security of the data, security and privacy, and also the international standards in this field, and the future directions. These are the 4 aspects that we'll cover in this second round. I'll first invite Dr. Shantanu to talk about the responsible AI and how data sharing should be guided by that.
Can I have the slides, please? Okay, so we'll be discussing in this session about responsible AI and data bias. Responsible AI is an approach for developing, deploying, and using AI systems that align with ethical principles and societal values. The aim actually is to create AI solutions that are technically proficient, socially beneficial, and ethically sound. AI should enhance human capabilities in decision making and not entirely replace human development. This is the problem that we always have. Can we replace human beings with AI? No, AI is not for replacing human mind. AI is just for aiding human mind. So what are the key principles? One is fairness and bias mitigation. I'll come to bias in a separate section, so I'm not dwelling into it here more. But, AI nowadays is everywhere. You travel from one place to other, Google Map, you can't live without Google Map today. You can't live without any damn thing in AI. Chat GPT, as was being mentioned by Dr. Shandar, right? Most of the important decisions are now being made by AI, to the extent that even hiring, and this is a big controversy, hiring is being made by AI-aided tools. So there can be bias which can sneak in. So how do we tackle this? We have to collect diverse data, so we can't restrict to a small place. And I'll dwell about this later a little bit. Algorithmic fairness, so how you have developed the algorithm is important. And most important is you have to have regular audits. Once you have made the algorithm and it is running, but then you are not auditing it, that creates a big problem. Then comes again transparency, and this is, self-explanatory. How do we tackle this? Again, explainable AI. What have you done? If you can explain that, right? Everything, every information should be there so that a person can log into that and get the procedure in which it has been done. The documentation has to be clear. Then visualization tools or graphs of other visualized aids which actually can help people understand how AI processes that information. Then accountability. When you do this, you have to have a person who is accountable. None of the methods that you employ will be foolproof. There will be mistakes. I was reading a guideline that was published in debated in US Senate, and that came out as a memorandum of understanding, and in that very clearly it was written, you do not attempt to make your system such that it can never be hacked. It will be hacked, right? It will be 99.9% it is safe, but that 0.1% is there. You need to have a person accountable who, if that 0.1% chance it is hacked or something happened, he can immediately take an action. So a person or a group of persons who has to be accountable for that. There should be feedback mechanisms. There should be ethical review boards from time to time. The other is privacy and security. This I have spoken while I was discussing ethical way of data dissemination. But one important thing is one basic principle. Just whatever the data is required for doing it, only collect those data. Do not collect XYZ data which you will never use. The more you collect, the more the chances are that it will be wasted. Right? And you have to have robust encryption. Still, Indian guidelines are there for all biology or human experiments involving human subjects. We should adhere to a HIPAA guideline kind of thing. And again, you should do a regular security audits. Now coming to bias, I was talking about bias. There are 5 different kind of bias. Now let us understand when we do the bias, and I'll give you small examples which has nothing to do with science. The first is confirmation bias. What is our belief? I'll give you a small anecdote. We all probably have heard this story. Dr. Sudha Murthy, when she lands in Heathrow Airport, she has to fill where she is going to stay. and she wrote 10 Downing Street. And the immigration officer looks at it, looks at her and said, "What? Are you joking?" Why? Because she was wearing a sari. A person, an old lady wearing a sari with a bindi cannot be going to a Prime Minister's residence. That is conformational bias. Think about it. You will get this in your day-to-day activity also. The next is historical bias. The data that we had, and we have been debating since last probably 7-8 months about the role of exit polls. Right? Exit polls are wrong. Historically, if you look at it, there were patterns by which people used to vote. Say about 30% of the women used to vote one point of time. 50% of XYZ community used to vote. Now, if that probably has changed over the years, but now, if you go back and use that algorithm, all your data is going to be biased. So, historical bias deals with historical facts which are probably not anymore relevant. Then comes selection bias, and this we all know, and we all do. I'll give you 2 examples. One, I want to do a project where I want to understand the biomarkers of cancer. How am I taking the sample? I'm taking the sample from the hospital. The clinician tells me which sample to take. Now that is biased. If you do not put a protocol like consecutive patient, then there is no problem. But if it is not consecutive patient, you need to write down what is the criteria of patient that you are going to take. It cannot be on the whims and fancies of the clinician that I can ask this person to give it. The second kind of selection bias. You want to do a study on the effect of alcohol on human health. And where do you sample? You go to the bars and nightclubs and do the sampling. That is selection bias. Then comes survivor bias. We all try to feel that people who survived are great, and that's why we try to remember them. Right? For example, if you are told that a person has not gone to a college but has become a big entrepreneur, it doesn't mean that to become a big entrepreneur you don't have to go to a college. That is survivor bias. And the next is the availability bias. If you are, whatever you're seeing just around you at that time, that is the availability. For example, if you look at the odds of plane crashing, if you ask anybody to do it, just after a plane has crashed, the odds will be higher that he will say that the chances are more. So those are availability bias. So we need to take care of all these before sharing data. Thank you.
Thank you very much. All these are issues that require a lot of nuances and perhaps a long time to discuss and deliberate. We are just hearing the keywords and trying to have a general kind of curriculum for us to understand the data sharing principles and the guidelines. I invite, in terms of the data security and how it is happening in IBDC, I invite Dr. Deepak Nair to share his experiences about if there are any incidents of data privacy, security breach or privacy and any of those issues quickly.
Right. Thank you. There are no untoward incidences so far, but this is a constant battle. There are always people trying to access, but there are ways and means to prevent them from, getting hold of data. And also much of the, so in case of IBDC, the data, as per the biotech pride guidelines, the data can be of 3 types. One is open access data, another is controlled access data, and the third is no access data. So from a user perspective, we are always thinking about the first 2 types. So the open access data is what people have deposited in IBDC, and it stays under embargo for a period of time until the researchers can hopefully publish their work, after which and whenever the depositor gives the go-ahead, it becomes open access. And the other data, like the human genome data, is under controlled access, which will only be accessible through a certain set of protocols and approvals from the relevant companies. So even in this case, this data is kept physically in different locations, and only what has been approved will reach an area where it can be accessed by the approved user. So there are clear guidelines from MIT on how to maintain security in data centers, and all that we are following now. And as advised by DBT, we are also getting regular security audits done to make sure there are no vulnerabilities in IBDC. Thank you.
Thank you very much, and again, congratulations to IBDCs for providing such a secure and robust environment for data sharing, and it is only growing. And no science and no technological concept can stand in isolation, so we have to look at what is happening in other countries. So what are the international standards? I'll invite Dr. Swapnil Narani once again to share with us about what is happening in this field elsewhere. So we put ourselves in perspective.
Can you put up the PDF again, please? Right, so internationally, there are a lot of things happening at various levels. India is a part of many of them. One of the first things that is happening is how consent is being taken. So since law does not allow us to take, needs explicit consent, it doesn't allow a blanket consent. There are some work happening towards taking it towards a dynamic consent, where the consent is not specific for one particular use case, but more broader use cases, which allows us to bypass the laws that are causing a problem in accessing the data. There is also a lot of emphasis on collaboration and having a lot of representation in all databases from across the world. Of course, that is not very easy to do. Even if you look at the TCGA datasets or the ICGC datasets, the proportion of Indian data in that is a very small proportion. So there's always going to be a relatively bias towards a certain population, that there is more representation of one particular population. But there is still an effort happening that we need to have a global representation in all the datasets. A lot of the privacy-centric frameworks are being evolved. Even our laws are changing, and there is. I talked about the consent and the dynamic, the broad consent model that allows data to be used for multiple use cases. Another thing that people are trying to do to overcome legal issues is use federated data access, where the data stays at its source of origin. Only computationally data is processed at the source of origin. Only the weights and the results of the computation are then transferred to the researcher. So the researcher tells what he wants from the data. The computation happens at the source, and only the results and the interpretations of the results are then transferred to the data source, to the researcher. Now, this has a lot of financial implications also, because then you have to create the necessary computing infrastructure to do this kind of implementation. The other advantage of this is that you can have many studies have shown that AI models which have trained on multiple data sets scattered across the globe using federated methods perform almost equally well as if on the other hand you had collated all the data set together. So these are all mechanisms that have been developed, are in process of developing. It is not yet widely accepted because it needs a lot of data source. We talked about the underrepresented non-European populations. Then open science and fair principles. So essentially the idea is that the world wants the data to be findable, accessible, interoperable, and reusable. So what we call the FAIR principles. And Biotech Pride is one of the examples of the policy that comes out of these FAIR principles. The idea is that the effort has been made that data should be available to other researchers for further research and to answer other questions which the original researcher may not have thought of. But as AI comes into play, it is very difficult to even imagine many of the ethical considerations. So for example, looking at normal X-rays, AI models can now predict the approximate year in which that person will die. Now, how that information is being processed is very difficult to understand. And therefore, an X-ray is otherwise non-identifiable. You cannot really identify a patient. But if you can predict directly the outcome of a person's life, 10 years, 20 years down the line, with reasonably high accuracy, that is going beyond what we can visualize as humans. There are certain things that we ourselves, as I am an AI researcher, I am a pathologist, I run a computational pathology lab, and we look at images to predict response to therapy. We look at images to predict genomic mutations. So we look at tumor images, and we can predict genomic mutations, at least in a proportion of cancers, with very high accuracy of 95% and above. So there are certain morphological patterns which are not visible to the human eye, which the AI models can look at. And these are things that we have not imagined before. And therefore, these ethical principles are going to change, and that needs continuous monitoring. And therefore, if you have seen even the public news, there is a lot of demand for stopping generative AI as of now in its current state, and then put in all the regulatory framework, although it has not happened, but there is a lot of call for that as well. Data localization I talked of, then there is the issues of monetization and technological innovation. So one of the ways of securing data is using more blockchain and more advanced secure technologies. Having said that, it is not easy to implement. It is not something that an individual researcher can do. You need an entire organization or the entire country to adopt it and go ahead with it. It is not something an individual researcher can do by himself. So the complexity of all of these ethical frameworks, legal frameworks, which change across geography, across disease types, across time, is so much that there's no single person who knows everything about all of this evolving scenario. So you need a lot of resources to know at that point what is the most relevant laws and ethical rules that you need to abide by, and how do I use these resources, computational resources, to really access this data within the ethical frameworks. So this is an effort called RDM Kit from the Elixir Europe, where they actually are maintaining a live collation of all the tools, the resources, the information about the laws, ethics, as well as the different software platforms, technology platforms that you can use to apply FAIR principles in your research. And also at the same time regarding the data sharing practices, how to share data responsibly, considering the ethical and legal issues that are different in different countries. You can even have, they also tell you how to make your data management plan, how to implement that data management plan, what all people you should have, what all technologies you can use, which all softwares you can use for accessing different types of data. And all of these tools are open source. So you can actually take all of them and implement in your own research to do all of this. Thank you.
Thank you very much. We are almost in time. I'll just ask Dr. Ritchie to give 1 minute and put it. Now we are talking about where we stand today and what we have been doing so far, but what is the future perspective, if DBT has any plan towards how data sharing will proceed, and any perspective of funding or policy?
Yeah, so they say that, people, human makes plans and gods destroys it, right? So as on date, we are coming out with these protocols which will be released in the afternoon and will be made available publicly. Again, these actually are the nuances for sharing of data, basically for a public funded research, and we do not, however, kind of dwell into, as on date, we do not want to dwell into, how we share the data for other communities, or probably we are right now also in discussion that how this data could be shared with the international agencies. We are trying to get into INSDC coalition. I think there has been already talks when they visited last time in India in March 2025. We're trying to be globally connected for other data sets like proteomics data set for like PDB, global PDB, and again metabolomics data sets where there can be a platform for global data sharing. And so that, and we are also in touch with the stakeholders where, the IBDC's accession number is kind of going to be acceptable for, we're making our efforts, best of the efforts, where IBDC's accession number may kindly be accepted by the publishers worldwide. So these are some of the steps we are taking forward, but again, we were trying our best and hopefully we'll get there someday. Thank you.
So thanks a lot to all the panelists. We tried our best to present to you a structured discussion, and it's time we can perhaps take one or two questions from the audience. We would have loved to have more question time, but I think we have already exhausted ours. Yeah, so.
Have a query regarding your dynamic or the consent taking. Now, when you take a global consent, which is what we've done for many of our studies, when you are doing a sharing data, which we will be doing for other objectives, that global consent would be valid, right? Because it's very clearly identified. Where would the caveats be there?
In law, there is no global consent.
That's the question.
It is if you look at the personal data protection laws, etc., there is no global consent. It is ethically we are on the right side, that we have taken consent for multiple use cases. So on the ethics side, we are there on the right side, but legally there is no law like that.
So how do we move forward, and particularly for funding agencies now, because there's a lot of data in these biorepositories, how will this data be shared if the law kicks in? Or if some participant or patient turns around and tells us, why are you using this data? So I think that's one point which needs to be flagged.
So one of the ways is that the right to be forgotten or withdrawal of consent, if that can be incorporated in that entire process. So the global, when you take a global consent, there is also a right to be the consent to be withdrawn.
Yeah, that's there in all informed. Yeah, that's there. That's
so
there.
Is one of the ways, because initially the patient has offered that consent. Now he wants to withdraw. Now,
no, but then that means going back to them.
The law kicks in once the complaint is raised.
Yeah, but you see, you're sharing data. We're not going back to the 12,000 participants or to the hundreds of participants who may be part. So I don't know. There is a challenge there, and I don't want to take up. Yeah.
We agree there's a big challenge there. It's not very clear. There's no clarity on that.
Yeah, how do you handle? Yeah, sure. It's not going to be easy.
It's a wonderful the discussion. I'm basically from pharmaceutical background. I'm looking at how this data can be useful for the regulatory perspective and the industry perspective. The objective can be refined in the better way for that, the translational aspect. This is the question.
So there is one thought about where we can, so regulatory agencies often validate certain products, certain models, or even regulatory agencies have started validating and approving AI models, at least in the US FDA. Now they would obviously need external data sets to validate the findings. So when the maker of the model or maker of the product has done the testing, they have done it on a particular data sets. Now these data sets will be different for different models and different this thing. Now the data sources like IBDC and other data sources can actually act as a service platform for the regulatory agencies, where they can offer that data for validation, where the model can be run on, let's say, IBDC resources or some other resources, and the data is not transferred to the regulatory agency. Only the results are transferred that, okay, this model performed this way on this particular data set. So this is one of the ways of also perhaps monetizing the data, which is ethically perhaps correct.
I would like to add, so in the translational perspective, for example, suppose there was some data personalized to some individuals and some drug was designed specifically to those types of individuals. Now this is a translational component who actually owns the rights, who actually produced this data, who actually generated the last mile product out of it. So these issues are still in the gray area, and there is a lot of thinking. That's why I said there are many nuances that we have to sit down upon here. Here we are basically talking about broader aspects of this.
This is a very serious concern. Analytics need to be done based on the hypothesis. Then who created the analytics? Benefit, how it goes to the analytics person, created person. Defined.
Hi. So we are talking about data sharing, but metadata is also quite important, because without it, the reuse becomes almost very difficult to actually do anything about this. So when we talk about clinical data, we also have to protect the privacy, and all these aspects come into picture. So how do we balance these two things?
Yeah, so that's a very important point actually. So you will see when we created this, we had a discussion on this. So your raw data and metadata are separate. So there is a committee which will evaluate why you need the data, what are you doing with the data, and what is the outcome of the data. There is a series of questions. The metadata will only be released case to case depending on what your question is. So everybody is not going to get access for all sorts of metadata, which probably is important. But metadata will be available, and there will be people who will judge whether the metadata can be given to somebody or not.
Just to add to this, again, we should not forget anonymization of data. Again, when IBDC is accepting no personal identifiers is being accepted, there's the data anonymization is the primary, even during the deposition of the data anonymization is the primary step there. So that has to be taken care of. Thank you.
So a case can emerge, for example, when you go to a doctor, your personal data is with the doctor. So in that regards, one may wonder whether that kind of data should be shared with those who can provide us better solutions. So currently this fine graining of the sharing rights, those systems are not in place. And can we have some clinical small group of people who can share this data and develop better solutions? So these things are perhaps still in the very early stage, and there are many difficult scenarios in that.
Very interesting concept that presented by Santanu about the biases, and the kind of biases that he given the example of, that comes from natural intelligence when we get biased because of the prior information that we have. But in artificial intelligence, I believe that there are scopes of de-learning and relearning, so that you can break the pattern and add new data so that new pattern can be developed. So my question is that, don't you think that this bias breaking AI will provide a solution in future?
Absolutely. And that is exactly what responsible AI was. One of the point was you need to evaluate, you need to learn from what the data was provided, and then correct the AI based, algorithm based on that. It has to be done on a continuous basis.
So thank you for fantastic session. I am Dr. Balendra from DBT. So
Like for other digital data, there are increasing concerns for genomic data too, especially in terms of lot of genomics driven market is in place, and it has a lot of future market potential. So how we can have national international standards? How we can have national international standards that really can have some mechanism in place for benefit transfer to the different stakeholders and including the local populations as defined in National Biodiversity Act and Biological Diversity Convention too. Thank you.
Yeah, so he's my fellow colleague from DBT and he's looking after the Biodiversity Act. So obviously I was expecting the question. So that's what I was, on the dais I was actually trying to refrain from saying this, but right now the guidelines do not delve into any kind of benefit sharing. Okay, this is mainly for the research purpose use only. I stopped my words over there actually when I transformed to INSDC chapter, but this is these guidelines and these we have Chairperson NB as the Interministerial Committee co-chair. So this was extensively discussed with him, and in the guidelines, when you read through these protocols as well, they are mainly for the research purpose. As on date, we do not well into any of the benefit sharing portion or any commercial angle as on date. So I'll like to rest my case here. Thank you.
Thank you.
Let me add one sentence. When we formulated biotech pride guidelines, NBA was very much there on the board, as well as Ministry of Environment was on the board. And when we have formulated these field protocols, the document through Interministerial Committee has gone, and on Interministerial Committee, DBT is the DBT Secretary is chair there, and for human data and towards medical sciences, we have DG ICMR as a co-chair, and for other biodiversity like issues, we have co-chair, chairman of NB. And we have a balanced composition of Interministerial Committee. Each and every stakeholder, including all the areas of biosciences, are covered under Interministerial Committee. And we have approvals, we have recommendations for biotech pride guidelines as well as field protocols. As Dr. Ricci mentioned, at present, we are going step by step. Initially, the data will be shared with the researchers. Then we will come up for the biodiversity issues. Then we will come up for the commercial angle. We want to have the experience, and we will go with the experience, and there will be questions always remaining there, but we need to have the answers with the experience. Okay, thank you.
Thank you very much for your intervention, Dr. Ninabe, and thank you again, all the panelists and all the participants for patiently hearing us. We don't claim to have provided you solutions. We raised the concerns and we raised the issues that we have, where we are, and the way to go. I'll end this panel with one couplet, because you may have been it may have been a very dry discussion. That says that तोड़ता कोई नहीं राह की दीवारों को, हम भी जाते हैं उन्हें हाथ लगा आते हैं. That is how we aim. We just go and touch these issues. We do our bit, and then there's a long journey towards our solutions. Thank you very much for all of to all of you.
Thank you, Dr. Shandar, for conducting this panel discussion. May I now request Dr. Kalevani Ganeshan, Scientist of DBT, to felicitate our panelists. May I request all the audience to please be seated. There are a few announcements to be made. Please be seated. The panelists are still on the stage. Okay, thank you. Okay. Dr. Shantanu Chaudhary. Same. Dr. Deepak Nair, please. Dr. Swapnil Dane. Dr. Richi. Thank you, ma'am. I request all the panelists for a group photograph. Oh, yeah, I'm going to announce. So there's an important announcement regarding lunch. The lunch is in the NXC building, which is adjacent to this one, in Hall A. All the participants are requested to carry their passes because security check is there at every step in this building. So you will have to carry your passes for entry into that hall. Only those who have passes will be allowed for the lunch. Lunch is in NXC building, Hall A, adjacent to this. Thank you all. Wait, batao.
We gather back at 2:30.
Okay. I request you all to gather back here at 2:30. Thank you. ऑनलाइन नहीं जा रहा है ऑनलाइन आवाज जैसा बिल्कुल नहीं आ रही है ये वाली तो बहुत प्रॉब्लम है हाँ ये बहुत प्रॉब्लम है आई डोंट थिंक सो मिनिस्टर उठ के बात करेंगे इसमें एक तो उनकी टीम को बोलना पड़ेगा हाथ वाला माइक यहाँ पे रखोगे ऑन करके उनकी टीम से बोलते हैं ये ट्राई करना पड़ेगा आपको ना रुचि ये ना ट्राई करना पड़ेगा लंच में चेक करवा लो अभी करा लेते हैं
हाँ हम दोनों। अरे हाथ ऐसे नहीं, मतलब माइक बोल रहे हैं।
प्रोफेशनल नहीं वहाँ हो गया ना,
बस वहाँ तो हो गया।
इधर ऐसे। अब आपकी चलो, अब आपकी चलो।
हो गया, थैंक यू।
नीतीश होगा, नीतीश। ये चेयर जो मिनिस्टर वाली चेयर है वो ऊपर कर दीजिए, बीच में। ये सारी एक जैसी कर दीजिए। हाँ ये ऊपर, वो नीचे से होएगा। और सबसे थोड़ा फिर भी ऊपर है तो वो बीच में रखो। मिनिस्टर वाली चेयर सबसे बीच में रखी है, वो बड़ी है उसके बीच में कीजिए, सबसे बड़ी मतलब बड़ी ठीक है, तो कर दीजिए। एक हाइट की है ना, वो देखने में जितनी छोटी लग रही है। अभी भी ये चेयर उसको थोड़ा नीचे कर दीजिए, या ये चेयर ही वो कर दीजिए, सॉफ्ट कर दीजिए, उसको बीच में कर दीजिए। नहीं बीच में कर दीजिए, वो अपनी चेयर सबसे ऊपर चाहते हैं, उसको रहता है कि मेरी चेयर सबसे ऊपर है। ठीक है, जो यहाँ पे आपस में बात करते हैं, बराबर एकदम उसको देखने में ऊंची इधर कर दीजिए, इसको इधर कर दीजिए, बात करते हैं, सारा टेंशन नहीं है। बस हो गया आराम से, इसको इधर कर दीजिए। ये सबसे ऊंची हो रखी है सर, बाकी सबसे बेस्ट है, इसको बीच में कर दिया। अच्छा अपने दोनों पहले आप वो चेक करो, IBDC चेक करो, कर्टन लगा के चेक कर लो एक बार, और वो पीक प्रोटोकॉल्स का बुकलेट जो खुल रहा है हम करेंगे।
Check 1 2, hello check, hello 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 check।
इसमें आवाज आ रही है जब लाइव फीड में जा रहा है ऑनलाइन, उसमें नहीं आ रही है। देखना भैया अभी आने दो उसको, मैं वहां पे और चेक करा दूंगा। अरे कभी भी ले लिया था रिची नीचे है ना। आ जाओ संजय। 2:30 तक लंच है। हाँ वो बस 2:30 के बाद लास्ट डिस्कशन होएगा 1 घंटे का, और फिर जब मिनिस्टर सर आ जाएंगे तो जस्ट स्टार्ट करते हैं। अराउंड 3:15 पे मतलब 2:15 2:30 के आसपास हम जब तक गेस्ट आ जाएंगे। ठीक है, जो इसमें सुमिता, जो तुमने मुझे वोट ऑफ थैंक्स दिया था वो जीएम पे मिल सकती है। चले आप लोग लंच नहीं कर रहे संजय? 5 मिनट सर, मैंने चेक कर दिया, कर्टन चेक करके कर लो लंच। तो आपको डिलीवरी बेस्ट तो नेक्स्ट सेशन में कौन सा
Hello check 1 2, hello check. Hello check 1 2 3, hello check. Check 1 2, hello check. Check 1 2, hello check. Hello 1 2, hello. Check 1 2, hello check. Hello check 1 2, hello check. Hello 1 2, hello check. 1 2, hello check. Check hello 1 2. 1 2, hello check. Check check hello check, hello check. Hello check. Check 1 2, hello check. Hello 1 2, hello check. Check hello hello. Hello check hello. Hello 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 check. Hello mic check hello check. Check hello check hello 1 2. Check 1 2, hello check hello hello. Mic check hello check. Hello check hello check. 1 2, hello check. Hello check 1 2, hello check. Check 1 2 3, hello check. Hello hello.
नीतीश को सुनाओ। अब मेरी मैं बोल रहा हूँ।
एक सेकंड सर।
चेक 1 2 3 चेक।
हेलो 1 2।
बस होल्ड करो, दोनों की आवाज़ सुनो।
देखिए सर क्लियर।
ये मेरी है, ठीक। अब आप
क्लियर आ रहा है सर? एकदम क्लियर आ रहा है?
सही है।
ठीक है भैया। हाँ चलो ऐसा है, केबल में प्रॉब्लम होती है कभी।
पोडियम पर सही आ रही है, उधर भी कम आ रही है, उसी में बोलना इसको उठाना उसको। क्योंकि वो कह रहे हैं इसका हैंडल का भी सही जा रहा है, पोडियम का भी सही जा रहा है, और 4 बंदों का जो है वो कम जा रहा है, ठीक है। अगर इसकी लेवल कम आ रही है तो उठा दे, क्योंकि ड्राइव में कम जा रहा है, तो उठा दे अपना लेवल, तो अपना ध्यान कोई बात नहीं। मैं
Genome India, unlocking the potentials of the Genome India data for public health. So Genome India is a visionary flagship project funded by the Department of Biotechnology, Government of India. This program was launched in January 2020 and recently completed sequencing of 10,000 genomes from healthy Indian individuals, spanning the length and breadth of the country. This Genome India, therefore, is an important step towards a healthier nation. Our next set of panelists will discuss how this valuable data set can be utilized to gain maximum benefits to revolutionize healthcare, empowering basic researchers and clinicians, leading to transformative precision interventions. So for the panel discussion, I would request Dr. Dinesh Gupta from ICGB. So please come on the dais. Dr. Madhulika Kabra from AIIMS New Delhi. Dr. Arindam Maitra, BRIC NIBMG. Dr. Devashish Das, BRIC ILS. And Dr. Mohammad Farooq, IGIB, New Delhi. I request Dr. Dinesh Gupta to moderate the session. Thank you, sir.
Yeah, hello everyone. Welcome back. I hope you have had a great lunch. And now we'll be delving into the discussion on unlocking the potentials of Genome India data for public health. And I have with me eminent panelists, and I will briefly introduce all of them. So towards my left is Professor Madhulika Kabra, who is Professor and Head Department of Pediatrics at All India Institute of Medical Sciences, and she has been recipient of very well-known awards like IC Verma Lifetime Achievement Award, IC Verma SIAMG Outstanding Scientist Award, and she has been chairperson of Center for Excellence of Rare Diseases Committee at AIIMS. She is member of Central Technical Committee for Rare Diseases, Government of India. She is member of National Apex Committee by MOHFW for planning a route map and monitoring of activities of the National Consortium for Research and Development on Therapeutics for Rare Diseases. Her areas of interest include intellectual disability, dysmorphology, storage disorders, newborn screening, and she has around 450 publications. She is working in the specialty of medical genetics for the last 30 years. Welcome, Dr. Kabra. And to her left is Professor Arindam Maitra, who is Associate Director, BRIC National Institute of Biomedical Genomics at Kalyani. His research interests include unraveling the transcriptomic diversity of OSCC tumor ecosystem. He is also deciphering the functional genomics of preterm birth in collaboration with other DBT institutes and other medical establishments. He is also interested in understanding gene regulation in human health. He is also was involved in genomic surveillance of SARS-CoV-2 and other pathogens. And he is currently project coordinator for NIBMG for India International Cancer Genome Consortium India project. And to his left is Dr. Devashish Das, who is the director of the ILS, DBT ILS in Bhubaneswar, which is a premier research institution under DBT. He is renowned for his contributions to life sciences, particularly in the area of molecular biology, genomics, and biotechnology. Under his leadership, ILS Bhubaneswar has made significant advancements in research related to human health, disease biology, environmental sciences, fostering innovation excellence in life sciences research. Dr. Das is also actively involved in various national and international scientific collaborations, aiming to translate research findings into practical health solutions. And to his left is Professor Mohammad Farooq, who is actually taking place of another panelist who was supposed to be here. Unfortunately, he is not here because of his illness. So he is also with us today. And by profession, he was a medical doctor. Then he did his PhD in medical research. And now he is very much interested in genomics of neurological disorders. So I think we'll make best use of all these panelists who are in this panel today. So to start with, what are the benefits of Genome India data, which is going to be released very soon by our Honorable Minister? So this release of population genome data has a potential to enhance public health by enabling disease risk assessment, personalized medicine, pharmacogenomics, and public health surveillance. It will aid in early intervention. It will also improve treatment efficacy. It will track infectious disease outbreaks and addresses health disparities, leading to more effective and tailored public health strategies. So there are several opportunities which are going to be unraveled by release of this genome data, many of which will enable us to delve into the research areas which were not possible earlier. It will also help us in formulating research questions which were not possible earlier and which are very much relevant to healthcare in India. So there are a broad range of topics to be covered. First and foremost is how can Genome India data improve the identification of individuals at risk for genetic disorders and population-wide screening programs, especially monogenic disorders. So I would like to ask Professor Kabra about this.
Thank you, sir, for a very kind introduction and for this very relevant question. So when we talk of, as a clinician, we were all looking forward to this data, particularly people like us who are working in the area of clinical genetics. So when we are talking about population-wide screening program or identifying individuals who are at risk of genetic disorders, we need to have some background in the country and experience in the country what we have with population screening as of now. So as was being discussed in the morning, we do have experience of newborn screening, which is a form of population screening only, but there we are looking for diseases basically. And good experience has been generated in the country through various programs, as mentioned in the morning, the ICMR program and the DST program. And I would like to specially mention the UMMEED program of DBT, which included aspirational districts also, and where we worked not only on newborn screening for 5 disorders, but we also looked at birth defects and included antenatal screening also for the mothers for certain genetic disorders. And through this, again, a large data set has been collated. Not genomic data, but we do have some targeted genomic data available because we did molecular studies on babies who were found to have disease. So this kind of information is very targeted, availability of biochemical data which is there, the incidence of hypothyroidism or congenital adrenal hyperplasia or G6PD, which is directly applicable, that you know that this is the incidence. But when we are talking of, and then we have now government-based programs like NEEV, which really has a large data set, so which is led by Dr. Seema Kapoor, and the UMMEED program was actually spearheaded by Dr. Suchita again. So the large data sets which are collated, which will give us some idea about the extent of problem. But then when we are talking about genomic data, which is now available through this project, it's a totally different ball game. So what we gather, and we will be hearing about the results, in this process, a large number of variants have been identified in the population, and these may belong to various categories, which could be autosomal dominant, recessive, and even X-linked categories. And based on that, it has been identified that a set of genes which are deleterious, like for familial hypercholesterolemia, even for breast cancer predisposition, all these are there. So actually speaking now, if we want to really apply it at the level of population screening, which is, and even we were talking about genomic newborn screening, which is far from in practice. So we really, what need to do is that now all these variants which have been identified have to be collated. So we need to have these variants which are there in the general population, which are at a higher frequency but of rare disorders, and then we classify them which are dominant, recessive, and all. And then it is also mentioned in the results that it has been identified that there are population-specific variants also. So there may be some vulnerable populations which are predisposed to certain genetic disorders. So I think this will be a work in progress where we will have to have this data set, the variants which have been identified, try to find out which are pathogenic and likely pathogenic, reclassify some of the variants which are novel, and it has been identified that the novel variants which have been identified through this project, actually many of them belong to the category which ACMG classifies as the deportable variants. So now we can have a data set where we have variants which are specific to Indian population and the variants which are there already reported worldwide. And if at some point in time we are thinking of population-based screening, the relatively commoner variants causing rare disorders can be considered for screening. And then the populations which have a very high frequency of certain set of variants which are pathogenic or likely pathogenic or are reclassified based on various tools, then that population can be sort of educated, made aware about their predisposition for that particular genetic disorder. So, I mean, it is evolving, but we are sure that with this data set, apart from population screening, we will be helping in a great deal to identify carrier frequencies, which we'll be discussing in the later part of discussion. Thank you.
Yeah. Thanks, Professor Kabra. That was very useful insights to start with. Professor Arindam, would you like to add more with specific to cancer genome screenings?
So I would thank you. I would just carry forward from where Professor Kabra mentioned. I would move forward to something cancer as also a part of as a larger picture of complex diseases. So as we finish off the low-hanging fruits of monogenic disorders, although now those things are also being reopened as more and more genome data is available, we work on complex diseases, which is the next challenge for the public health in front of us. And I'm not going into the details of the names. All of you know about the complex diseases that we face as a population or as a country. So we are dependent on a large amount of genomic data, population genomic data that should be made available for us to conduct this kind of research, identify variants which can go into predictions or which can go into intervention measures, development of intervention measures. Now, when we do that, we invariably depend upon population databases. And when we go to these population databases, we find that most of the data belongs to a small minority of the populations worldwide. Asian populations, particularly South Asian populations, contribute the biggest amount of number of human beings on this planet and also shares the biggest burden of complex disorders. But we do not have enough genomic data there. And that's how this whole Genome India program was conceived, to address the problem of Eurocentricity, bringing in Indian population data, genomic data into this unveiling. So as we stand today, we are on the brink of a very promising times for disease genomics research in India. It's very exciting. For a disease genomics person like me, I'm excited with what is now in front. Simply, I will give you an example with which I work. So in the morning, Professor Shinjini Bhatnagar talked about the Garbhini program, where we are looking at a very heterogeneous, complex trait of preterm birth outcomes. It's a birth outcome, adverse birth outcome, it's called preterm birth. Now South Asia contributes the largest number of preterm deliveries every year globally. And we know the associated public health problems that are associated with preterm birth. She mentioned very nicely in the morning. When we conducted the first maternal GWAS on preterm birth from South Asia, reported till date, on more than 6,000 women at this point of time. And we did that using standard workhorses of GWAS, which is heart content microarrays. And we used probably one of the best globally optimized heart content microarrays that are available at this point of time, which looks at about 700,000 polymorphic markers. Now, when we did it on our population or our GWAS participants, we found that about 50% of these 700,000 markers in the array are not informative in our study participants. They are either not polymorphic, or their polymorphism levels are so low that we cannot use it for association studies. So these kind of challenges, and I'm sure there would be other people in the room who would be also facing similar challenges with other complex diseases, sitting in India, working on Indian populations where there is a high burden of these diseases. Now that has made some of us move away, and this is how the globally genomics research is going to move, is from these hard content cost-effective genotyping measures to more genome profiling based methods, which will be able to capture population specific variants and which will be more cost effective in terms of doing it as a screening or as a profiling measure across large number of samples, because complex disease research will depend on large sample sizes. There is no getting away from it. Now, as we do that, we have to go back to the population genome data to capture the entire genome, because high depth whole genome sequencing is out of the question. It's not possible in terms of finance or resources to do that. Now when we do that, this is where Genome India data is going to come to help. So far we have been struggling with at best 1,000 genomes data, where there is a limited Asian content, Indian content, and little improved with Genome Asia data and some other population data sets that had been released, but they are very small in number. We are absolutely confident now that with the Genome India data being there, with 10,000 individuals per genome profile, we'll be able to combine this with the low-cost genome profiling methods like low-pass sequencing approaches that we are working on right now in most of our studies. And we would be able to impute the genotypes and make this a very effective solution for our research. So we'll be able to capture the missing genotypes so as to see the missing genomic component in most of the studies that have been conducted till date. And ultimately, this will lead to methods which will be useful for doing and feasible to do population screening. So this is where we stand at this point of time, and I think these are very exciting times ahead.
Yeah, thank you, Arindam. That was very useful insights about and very shocking results that our population genome sequencing should be done more aggressively actually in order to bring out more fruits from this kind of sequencing projects. As far as I remember, that UK Biobank actually collected data sets of about 50,000 individuals. And that was for a country which is very small as compared to India. So this again raises the example and the need for such a large scale population-wide genomic studies. So Arindam, I would also like to ask you, apart from screening, how can Genome India project outcomes help in personalized medicine or personalized treatment?
So that's what, I mean, once we use these Genome India data to leverage it to our advantage, one is identifying and getting more population-specific variants. We are also having, this is like a, kind of a super control. So we have this whole data set of 10,000. So whenever we are even doing studies where there are rare variants, we are using exome or other kind of sequencing-based approaches, identifying. So this 10,000 genome data is going to be a useful database to screen out and identify variants which are likely to be pathogenic, which will be, of course, subject to various verification measures, and those will have a direct path to the clinic. So I think this is how this whole Genome India data will result in a more rapid transformation of our genomic discoveries in diseases to clinic and practice.
Thank you, Arindam. My next question is again to Professor Kabra. How can Genome India project help in better prenatal genomic testing to improve maternal and child health?
Okay. So what has happened over the last few years is that the genomic science has very rapidly moved from bench to bedside, which was actually not happening earlier. We had somehow clear distinction between this is research and this is clinical. But then with the exomes and genomes being available all around, this is very frequently being used not only for postnatal diagnosis, but also for in the prenatal situations. So here the issue is that, again, I'll just take the issue of the population screening, and we talked about the screening, antenatal screening, which was initiated under the Ummeed project. So now what is happening is that in the antenatal screening, there is a concept of screening for certain common disorders like say thalassemia in our country, and now also for spinal muscular atrophy. But now there is a move to offer screening to the couples that they may get their exome screened. So like a couple exome, and then maybe panels also for specific populations. So now this has not been accepted for a low risk, so-called low risk population, but there are studies available even from India that in low risk populations also deleterious genes and parents have been found to be carriers of certain genes. So now with the high risk screening, so what we are following in clinical practice is that we are screening couples who have high risk. There are certain criteria for being high risk like family history, previous baby affected with a genetic disorder and so on and so forth. So here we offer couple exome screening and try to find out whether they are sharing some common recessive genes which makes them susceptible to have an affected baby, or a X-linked gene which the mother has. So particularly in consanguineous couples, we have found 7-8 deleterious genes in the same family and both parents being the carrier, and then the counseling becomes very difficult. So now with the availability of Genome India data, and what happens many times is that we get a variant which is so-called variation of unknown significance, whatever data we have from the global data availability, and we really don't know whether what significance it has for our own country. So this, if we have a data set of our rare disease variants, then it may be useful to classify these. So this would be one important area where we can probably screen better and we can find out and interpret that better. The other very important thing, which is I think through the this data which we can have, again, if we have a set data and reclassify the variants and find out which are our novel variants and are they pathogenic or likely pathogenic, because many times what is happening now is that we are doing fetal exomes primarily in situations where the family is already having a disorder or suspected disorder, but in situations where even in the first pregnancy we find something in the ultrasound that the baby is abnormal, and we end up doing a fetal exome. And then we find out a variant which we are not able to interpret and relate to the disease. So again, if we have a data set which is India specific and we know it is common in our country and maybe it is a pathogenic or a likely pathogenic variant, so it would be easier for interpretation and counseling the parents for having a reproductive choice, for continuation of pregnancy and so on and so forth. So based on this, as is there the recommendation of the ACMG guidelines, where they are saying that they recommend screening in the antenatal period primarily based on the frequency of particular variants in particular populations. So they would say this is a set which you apply for the whole population. As I gave the example of thalassemia, SMA, maybe DMD, Fragile X also. But then there is a set of genes which are to be screened in, say, Ashkenazi Jewish population. So then we, in fact, in our country also, there are certain populations which have a set of genes for which the carrier status is extremely high. So we can have such panels available also, and these can actually be used for screening in couples, at least as of now in high-risk couples it can be applicable. So I think these would be two important applications as far as clinical applications in the prenatal, antenatal, preconceptional setting is concerned. Thank you.
Thanks, Professor Kabra. This once again reiterates the importance of the outcomes from the Genome India project and how we can use it for developing more diagnostics which are appropriate to our prenatal screening and postnatal screening in Indian population and improving our health services. So my next question is to Professor Debashish. How can we implement artificial intelligence to genomics data sets to improve Indian health system?
Thank you. Being a computational biologist, it's an exciting time for all of us. The field that generally people think of parasites, but these parasites actually do not do this analysis for self-survival, but rather convert the data to some meaningful knowledge, and then with the help of the domain experts, that gets converted into some wisdom. Having said that, 10,000 genome is an exciting time. Lots of data are going to be available to the data analysis. AI exactly is not needed at this point of time, but ML and deep learning are two methods that will be highly useful to analyze this kind of genome data that we have. For example, we will have several thousands of mutations or polymorphism available from this data. And many of them will be demarked as that variant of unknown significance. Why unknown? Because nothing is known so far, but can we predict something? In the audience, if there are computational biology, you would have heard of some tools called deep sea, demining, or for that matter, expecto. These are some of the deep learning tools which takes the variant information or epigenetic information, gene expression information put together and then give some meaning to these variants and then predict the disease. There is something also which we think that the genome data that will be made available, we can do something called disease forecasting. There are mutations that is known and people can tell that, okay, this disease is running in the family and you can assign. But what about certain mutations or certain variant that has come up? Can we forecast that this will lead to maybe a neuronal disorder or muscular dystrophy or something similar? Because similar informations or pathways, genes are already known to be involved in those kind of diseases. So I think deep learning will play a lot of role when this kind of data is made available to computational biologists, the students who are started learning how to develop algorithms, they should develop new algorithms based on this data. And I think the medical fraternity will take the benefit of that before it is reaching to the public. I think this is where hand-holding has to happen between the doctors and the computational biologist or the experimental biologist, so that before it is given back to the public who has given their precious sample, we should get some inferences out of this data.
Thanks, Debashish. To add to this, I would like to add that the project called IBDC project by DBT has systematically collected lots of data. And the ultimate aim is to develop a portal which will enable AI-based algorithm development. In this direction, we are already collaborated with ACTRAC, who are generated a lot of cancer-related image data sets and genomic data sets. And we are developing actually an exploratory portal which will enable AI-based algorithm development. And similarly, for other data sets also, there is a plan to come up with a omics toolbox which will enable such kind of algorithm development, which will have AI-enabled algorithms which can be trained using the data sets which are deposited to IBDC. So thank you, Debashish. My next question is to Dr. Farooq. Dr. Farooq, what are the implications of Genome India project outcomes for neurological disorders? How it can enable development of diagnostics and treatment plans for neurological disorders?
Thank you, Dr. Gupta. So technically speaking, so much has been said about like the benefit of having this data, like diversity assessment, diagnostic assistance, then risk predictions, pharmacogenomics. So having said that, in continuation of Dr. Kabra's this thing that they are helping in diagnosing monogenic disorders, estimating the carrier frequencies. So we know that like most of the genetic disorder, they have a pediatric onset, like almost like 70 to 80% genetic disorders are pediatric onset. But we often forget adult onset genetic disorders. So chiefly the area where I have worked in, like say, hereditary ataxias, where a person walks with a imbalanced gait. Other than that, there is a monogenic component of Alzheimer, monogenic Parkinson disease, monogenic motor neuron disease. There, what happens that, like say, for a very penetrant mutation, a very penetrant variant may got selected out because of the reproductive loss of benefit. But the adult onset disorder, the variant will have a frequency in the population. So definitely for establishing a diagnosis, you require a baseline reference data. And here the reference data would be the frequency. So technically, if you follow the ACMG guideline, there is one very important criteria which talks about that you estimate the frequency of a variant in disease group versus control group. So in anywhere, if your frequency in the control group is anything above 5%, or even you can say a common rare variant, maybe above 1%, you try to classify that into a variant of benign significance. So this is very important data, and we have covered almost like entire of the genome, not only the single nucleotide variation, which we know that they are the chief causalities for monogenic diseases. We have covered this CNVs. It will allow us to cover tandem nucleotide repeats, which is totally underexplored. There are a lot of adult onset neurological disorders which are caused by tandem nucleotide repeats. So there are definitive penetrative length, which we know that causes a penetrant disorder, but these repeats in low abnormal range, they are the risk factor for various adult onset diseases. So I think having covered the genome at this scale, it's a significant step, and I think I would say that possibly having an opportunity, being fortunate enough to be part of the Genome India team as well, I look forward for like expanding this effort to reach out to the other part of the country as well. And this is regarding the one aspect I mentioned. The last part also I would like to slightly highlight upon. See what is happening, like say even I've got a chance to work on this front as well, meaning the diagnostics of rare diseases as well, and the research aspect. So what do we see? Typically any monogenic disorder, the whole exome sequencing based yield is 20 to 30%. What do we see here? Because of this lack of availability of this population frequency data of variations, we are not able to classify our variants. At that number, that percentage is significant, 20%. So the 20% portion of any exome effort is diagnosable. But having unavailability of this data, we couldn't make the diagnosis. Suppose we have a lot of commercial entities being outrightly offering genetic diagnostics, but only 20% they are able to deliver or to say that they are actually a pathogenic variant. Otherwise, almost 20 to 30% time diagnoses are interpreted as VUS. So technically, it's going to help a lot. And pharmacogenomics again. So even I would say that many of the variants, we have our own variants, which we do not share with any other international population, any international database. So here, a lot of research is also required, not only for rare genetic complex diseases, as well as the drug response. So what do we call that? Like say, so drug response in terms of efficacy, in terms of adverse drug reactions. So we have an example. I was part of one study that predicted the adverse effect of 5-fluorouracil for it is I think precisely given for colon cancer patient, and the adverse event can be fatal. So what did we identify? That in the pharmacogenomic databases, there are 15 DPYD variations, which are supposedly called like the risk variants for 5-fluorouracil toxicity. When we checked our Indian population, we could find only 4 variations, which were informative. Remaining all 11 variants, they were all monomorphic. So what it is, like other variations are not there, or meaning can there be more variation in DPYD? So this sort of data will allow us to dive deep into that, say suppose that segment, say DPYD gene, identify more rare variants, deleterious variations, and predict effectively the drug response. So I think this is what I would like to.
Yeah, thank you, Dr. Farooq. You have raised a very pertinent issue of rare diseases, for which we have, unfortunately, very little data about Indian population. Some databases there, but yet a lot more needs to be done. So, Professor Kabra, can you elaborate how Genome India project data can be used for diagnosis and management of rare diseases in Indian population?
Yeah. So just taking forward what Farooq started. So one very important challenge which we have been having, because you are, as you are aware, the National Rare Disease Policy has been declared, and we are struggling with the definition of what is rare in India. So that is a challenge, and in fact in the policy the definition has not been spelled out as yet. Only a set of disorders have been included. So, and we have been repeatedly asked by the ministry to provide the prevalence or incidence of certain rare diseases. And how do we say it is rare? So I think with this Genome India data, as Farooq also touched upon, based on the carrier frequency of at least certain rare disorders which are as of now treatable, if we get the carrier frequency of these disorders, we can calculate indirectly the prevalence or incidence of these rare diseases. And based on that, we would not only be able to maybe define what is rare in India, but then in addition, and then we will have to have our own definition because all countries have their own definitions. But then we would also be able to identify the magnitude of problem. So suppose a disease which is rare, but the treatment is available, and we know that this is the possible prevalence in our country, then we can probably tell the government then, okay, this is a disease which is treatable, and this is the number which we have, though the cost is high, what will be ultimately the annual cost of treatment. So I think with this data, and this we have requested in the meetings also which we have been attending for Genome India, that if carrier frequency of rare diseases can be taken out as a data set I was explaining earlier, this would really help us in getting out the prevalence of rare diseases, which are extremely important to for the policy and for the benefit of the patients as well. The other thing is, which I again said, and Farooq also touched upon, that even in the best situations when we choose the families for exome sequencing or an individual for exome sequencing in a clinical setting, if we choose very judiciously, the yield may not go beyond 40% or 45%, and the rest are undiagnosed, or we end up with variations of unknown significance, despite the fact that the phenotype may be matching, but we may not have any other evidence for proving it, say biochemical, and we may not be able to do functional studies to prove that. But if we have a data set which we know, which is unique, because in the global data set, Indian representation is very low. If we have a unique data set for our own country, and we know these are pathogenic and likely pathogenic, we can relate it to the disease and confirm the diagnosis of rare disease, which is extremely important. We may not have treatment available for all, but now we have treatment available for many disorders. And in the same continuation, if we find a rare disease in the country, and then we find that there is a hotspot in a particular gene, and majority of the individuals have got that pathogenic variant only, one can work upon therapeutics also, because now we know therapeutics like gene therapy, CRISPR-based therapies are being done. And then we have been working in this area, like sickle is easy, there is only one mutation and you can work on that. And there are certain other diseases also in India, we have one predominant mutation. So if we identify that, it would be easier to work in the area of therapeutics as well. So that's from my side.
Yeah, thank you, Professor Kabra. So Arindam, how do you think the information from Genome India project will benefit the research in polygenic diseases?
So I will give you an example, again drawing from the preterm birth GWAS that we did. When we started doing it, we found that there are quite a large number of large European GWAS conducted in European women, basically. And we constructed the polygenic risk score. So for complex disease research, polygenic risk score is the work tool that you generally do to predict. And we found that when you use that on our data set, on the Indian women data set that we generated here in the Garbhini cohort, we found that these PRS, polygenic risk scores, fail to predict birth outcome. So now with data like Genome India, which is our own population genome data available, we will find more and more of situations where in complex diseases we'll have polygenic risk scores which will be able to predict complex disease traits more effectively. Whether that is to triage individuals, to modify their risk, you cannot modify the genes, but if you know that there is a risk prediction, then there is a risk, you can modify lifestyle and thus modify risk. You can also think of identifying genes, you'll be able to think of novel ways of intervention. In many cases, there are no interventions worked out, or more improved intervention methods which are more personalized, which are directed to the genomic profile of the individual, which will be more effective. So this is how I find that the Genome India data will ultimately benefit the polygenic risk.
Yeah, thank you, Arindam. So I think Minister is going to arrive very shortly. So in the meanwhile, I would like to put a question to all the panelists. So from the outcomes of this Genome India project, what do you think we can get the cues for the future projects which will involve population genome sequencing? Debashish.
Yeah, I'm reminded of a program that was that started in IGIB when Dr. Conclave was director, that called GoMed. That program directly gave the benefit to the patients when the recommendations came from AIMS or other doctors to look at the genomic profiling of the patients. In current day, I think this will double or triple the benefit, because now we will have a background population of 10,000 individuals, and the genomic information that we'll get from the patient can easily be translated to something actionable or something towards the disease. Farooq may add more onto this, how the GoMed program, according to you.
Yeah, exactly. I would add further that, like, say, it allows us to, like, say, immediate application of designing panels. Basically, as Professor Kabra said, that, like, we know there is some sort of a hotspot. So maybe in a given position, there is a hotspot. Then there is also over prevalence of a disease in a particular area or geography. So then you can really adjust your resources such that, like, you can divert your resources where the need is really been there, and as well as the maybe a geographic specific panel, or maybe reducing or making the low cost diagnostic, it is really going to help this way.
Yeah, thank you, Dr. Farooq. Same question to Arindam. What do you think are the cues that we can get from Genome India project?
So one is so this whole exercise is going to democratize genomic information. So and in addition to the cases or the examples that we talked about, I think itself population level data will also provide further information on many areas of biology which earlier we are not able to do in sitting in this part of the world with our own population. Thank you.
So thank you, Arindam. It is clear that integrating Indian population genome data into public health is essential for personalized healthcare, disease prevention, and addressing India's unique genetic diversity. The Genome India project aids in early identification of genetic disorders, supports population-wide screening, and enhances personalized medicine. It informs public health policies and lifestyle recommendations, improving maternal and child health through better prenatal screening. And however, there are many challenges, including ethical challenges that include privacy protection and equitable access. Responsible data use is crucial, requiring strong governance, which IBDC is committed to abide by. The data can enhance research, aid in rare disease management, and develop AI tools for public health. And comparing it with international data sets, it can provide valuable insights into global and regional health challenges. So my question to all the panelists is, again, do we need another Genome India project? Kabra.
Maybe with a larger number, because 10,000 genomes may not give us all the information, particularly for the rarest of the rare variants. So maybe a larger sample size.
Arindam.
Yes.
So two takes. One is we have to increase numbers. We have to profile more individuals in our population, and we have to, using this 10,000 data, we can actually use for other cost-effective genome profiling techniques and combine and reach out to larger number of populations that we can genotype. This is one. And second, overlying on this genomic variant data, we have to now look at the epigenome. And now we have methods established where we can actually generate the data and extract both genomic and epigenomic information. So I think these are the two areas where I think we should go
Devashish?
for.
My take would be we should do multi-omics rather than just increasing more and more number on the genomic side. We should do genome, proteome, metabolome, and epigenome, all these things. Like the CSIR cohort project is going on all across the country, 10,000 individuals. Similar such effect should be done so that, all this data can be combined and more meaningful outcome can be taken out of multimodal analysis. That's my take.
Yeah, great. Dr. Farooq?
Yeah, I would definitely say that, like, we should be expanding our efforts, and we can encompass more of medical genomes, where, like, say, not only the population genomics, but the genomics oriented towards phenotypes, that has to be also covered well. And I think more and more subject allows us to really capture the the rare variants, basically. So common variant, we might saturate after a point, but for obtaining rare variants, the real rare variants, because technically in every meiosis, we are generating de novo variations as well. So your singleton issues are there. So we need to really cover rare variants, and really, for that reason, more helps, actually.
Yeah. Thank you so much. And I also thank all the panelists for their valuable comments and participations. And it is, in the end, it is clear that there are clear and evident benefits from this Genome India project. The outcomes from this project can be used in improving health in India, and also formulating future proposals which will be more targeted and useful for Indian health system. So thank you all. Thank you very much.
Thank you, sir, and thank you, panelists. Now request Dr. Kakali Das Gupta from DBT to facilitate our panelists. Ma'am, please. डॉक्टर काबरा मैम आप मैम आप उस तरफ खड़े होइए, फिर यहाँ से आप अभी तो दीजिए, नेक्स्ट इसके बाद आप बोल रही डॉक्टर अरिंदम मैत्रा. डॉक्टर देवशीष दास. Yes, yes. Dr. Mohammed Farooq. Group photograph. I request all panelists for a group photograph.
Secretary and it's Dr. Suchita to join on the stage. Request the dignitaries to join on the stage. चल मैं जाता हूँ.
आप अनाउंस कर रहे हैं.
Thank you, Abhishek. Thank you, panelists. Now we move to the keynote session. Honorable Minister will be arriving any moment, and then we'll be starting our keynote session. Thank you. एक सेकेंड, पीछे मिनिस्टर सर जहाँ जाएंगे या ना वहाँ पे फोटोग्राफर भेज दो, आप चले जाओ। हाँ
दिख जाएंगे।
Honorable Minister has arrived at the building. He'll be here any moment. Thank you.
The Honorable Minister has reached. I request all of you to kindly keep your mobile phones on silent mode.
Good afternoon and welcome to the keynote session. I welcome our Honorable Chief Guest, Honorable Minister Dr. Jitendra Singh Ji, who has made time from his busy schedule to make it to this Genomics Conclave. Before we start this session, I would request Dr. Rajesh S. Gokhale, Secretary DBT, to kindly felicitate Honorable Minister Dr. Jitendra Singh Ji with a shawl and a potted plant. Now may I request Dr. Rajesh S. Gokhale, Secretary DBT and DG BRIC, to kindly felicitate Honorable Professor Ajay Kumar Sood, Principal Scientific Advisor to Government of India, with a potted plant. May I request Dr. Suchita Nenave, Advisor DBT, to kindly felicitate Dr. Rajiv Bal, Director General ICMR and Secretary DHR, with a potted plant. I also request Dr. Suchita Nenave, Advisor DBT, to kindly felicitate Dr. Rajesh S. Gokhale, Secretary DBT and DG BRIC, with a potted plant.
May I request Dr. Richi V. Mahajan to kindly felicitate Dr. Suchita Nenave, Advisor DBT, with a potted plant. May I request Dr. Richi Mahajan to kindly felicitate Dr. Arvind Sahu, Executive Director, RCB, with a potted plant. I request Dr. Richi Mahajan to felicitate Dr. Y. Narahari, IISc Coordinator, Genome India Project, with a potted plant. Thank you. Now may I request Dr. Suchita Nenave, Scientist G and Advisor DBT, for her welcome address.
Namaskar and good evening to all. We all are gathered here to participate in the landmark event, Genomics Data Conclave. It is indeed an honor for me to extend warm welcome on behalf of the Department of Biotechnology to all present here in person and in online mode. First and foremost, I express my deepest gratitude to Mananiya Mantri Ji, Dr. Jitendra Singh Ji, for his kind presence to grace this occasion. Hearty welcome to you, sir. Last year in February, Honorable Minister announced the completion of whole genome sequencing of 10,000 individuals, and it was committed that we will make the data available to our Indian research community very soon. Sir, today we stand this day to fulfill the commitment made. Gracious presence of the dignitaries on the dais, Professor Ajay Kumar Sood, Principal Scientific Advisor to the Government of India, Dr. Rajiv Bal, Secretary DHR and DG ICMR, Dr. Rajesh Gokhale, Secretary DBT and DG BRIC, is indicative that the promotion of science is a priority to them. It is my privilege to welcome you, sir, to this event. I extend my warm welcome to Dr. Arvind Sahu, Executive Director, RCB, and Dr. Narahari from IISc Bangalore. I welcome government officials, directors of the institutes, medical and scientific fraternity, my colleagues from DBT, friends from media, and all gathered here to this important event. Today we have gathered here to celebrate the success of Genome India project. Genome India data will help our researchers to find Indian solutions for Indian problems by accelerating research and development in different fields of biosciences. However, it is equally important to establish comprehensive protocols for data sharing to ensure judicious use, integrity, security, and reproducibility of the data. Earlier, this department has issued Biotech Pride guidelines. Now, under the ambit of these guidelines, the framework for exchange of data protocols has been formulated to establish a robust mechanism for data sharing and access. With completion of all these processes, we are now ready to make Genome India data accessible to our research community in a fair manner, transparent and responsible way. On behalf of the department, I humbly express my profound gratitude and happiness to Mananiya Pradhan Mantri ji. He kindly consented to deliver a special video address on this occasion that will strengthen our commitment for advancement of science in the country. Once again, my warm welcome to all the participants. Thank you.
Thank you, ma'am. Genome India is a government of India's commitment for a healthy India. Now I request Professor Narahari to kindly give us early insights into the Genome India data. Sir, please.
Honorable Minister of Science and Technology, Dr. Jitendra Singh ji, sir, and all the distinguished dignitaries on the dais, and all the learned audience. The Department of Biotechnology gave us an extraordinary opportunity to serve the nation by handing over the Genome India project exactly 5 years back. I would like to have the slides, please. It was exactly 5 years back that the Genome India project was conceptualized, and it was initiated. And through the dedicated efforts of the entire Genome India team, I think we have been able to achieve a few outcomes which are going to be like a watershed in genomics in the country. I would like to fondly recall the proud moment of 27th February 2024 when the Minister of Science and Technology, Dr. Jitendra Singh ji, announced the completion of the whole genome sequencing of 10,000 samples. Not only that, he also inspired us to do much more than what we have done. In terms of outcomes of Genome India, over the last 5 years, we have been able to establish a biobank of 20,195 samples for future research. We have completed the whole genome sequencing of 10,000 plus samples. Most importantly, we are ready with the digital public goods, and we have archived the sequencing data at IBDC. And we would like to consider Genome India as providing a pathway to genomics-based healthcare. The insights from the analysis have been extremely promising, and we expect that these findings will provide the scientific leads for affordable India-specific diagnostics and precision medicine. I would like to take you through a few early insights that we have been able to obtain with the analysis of Genome India data. To start with, this picture shows the allele frequency spectrum for as many as 130 million variants, of which 53.15 million variants have at least 3 minor alleles. Using this, a reference haplotype panel which was created, has been found to have a very high level of accuracy, even at minor allele frequency bins. This is going to be tremendous for coming up with imputation panels for cost-effective diagnostics. Then in another early insight, we have found that of the 20,000 or so genes, 5,700 plus genes have at least one loss of function variant. And these variants are both in the form of single nucleotide variant and also insertions and deletions. And we have found that all of these are high protein sequence altering variants, which is a very significant finding. When we look at an admixture plot of the Genome India data, it reveals tremendous genetic diversity in the Indian population. One of the major findings of the Genome India project has been to unravel the diversity that is very characteristic of India. And so you can see the 9,700 vertical lines partitioned into various GI population groups. The colors of vertical lines indicate different ancestries. And if you look at a principal component analysis of the Genome India data, this actually reveals 5 ancestries. And you can again see the kind of migration patterns, admixtures, which are characteristics of the Indian population. On the pharmacogenic genomics landscape, the insights that we have obtained, they're already yielding very valuable insights into how how people are going to respond to intake of drugs and so on. Okay, the data analysis that has been conducted so far also has led to the development of a Genome India database platform with which researchers, clinicians, and policymakers will be able to do genetic analysis and also planning. So with all these early insights and with the promise that we have of obtaining even more insightful observations, I think we have to look at the next frontier for the Genome India project. In some sense, the Genome India project has placed us at the cusp of a genomics-based health revolution in India. It has provided a pathway for understanding India's genetic diversity and has created a solid foundation for future studies. And the next frontier has to provide a comprehensive coverage for all linguistic and ethnic groups. A variety of healthcare settings and diseases should provide coverage to all tribal, rural, and underrepresented populations, and a coverage for all states and union territories of India. I think longitudinal studies are also required for a detailed understanding. I think for a country like India with a population of 1.44 billion and more than 4,600 population groups, I think the best way forward would be a 1 million genome program, including very specific disease cohorts. I think that would be a crucial step forward. Of course, this will call for such a mega project will call for highly distributed and well orchestrated operations, new funding models, and wide-ranging interdisciplinary collaborations. So with this, for providing this extraordinary opportunity for all of us, and also for the tremendous support that we have received, we would like to express our deep gratitude and grateful thanks to our Mananiya Pradhana Mantri ji, the Honorable Prime Minister of India, Shri Narendra Modi ji. We would like to profusely thank the Honorable Minister of Science and Technology, Dr. Jitendra Singh sir. We would like to thank the Principal Scientific Advisor to the Government of India, Professor Ajay Kumar Sood. We would like to thank profusely the Secretary of DBT, Professor Rajesh Gokhale. Many thanks to the members of the TMAC, Technical Monitoring and Assessment Committee, Professor Padmanabhan, Professor M.R.S. Rao, and Professor Partha Majumdar. And many thanks to Dr. Suchita Rinave, who has been tremendous in her support of this Genome India program, and all the DBT officers, Dr. Richi Mahajan, Dr. Kakoli, Dr. Onkar, Dr. Amit Tripathi, for their excellent facilitation. So to conclude, I think many thanks from Team Genome India. Many of them are here. Many thanks from all of us for the marvelous opportunity that is provided to us to serve the nation. We hope that Genome India will inspire the next generation of genomic innovators and herald a genomics-based healthcare revolution for India. Thank you very much.
Thank you, sir, for the early insights. Biological data sharing, especially national resource like Genome India database, is essential for tackling multi-dimensional challenges of modern biology. However, at the same time, proper data management and governance are critical for maintaining the integrity and reliability of the data. For the purpose, feed protocols framework for exchange of data have been formulated by Department of Biotechnology under the ambit of Biotech Pride Guidelines 2021. To shed light on the same, I would request Dr. Arvind Sahu, Executive Director, Regional Center for Biotechnology, to make a brief presentation. Please.
Thank you, Richie. Good evening, everyone. Dignitaries on the dais, Honorable Minister of Science and Technology, Dr. Jitendra Singh Ji, Honorable Principal Scientific Advisor, Professor Ajay Sood, Secretary DHR, DG ICMR, Dr. Rajiv Bahel, Secretary DBT, Dr. Rajesh Gokhale, Advisor DBT, Dr. Suchita Ninave, Dr. Narhari, scientists from across India and from various institutions, and the media people. So I'm standing before you to tell you a little bit about the feed protocols. And since we do not have too much time, I'll just rush it through this. So can I have the slides, please? So the feed protocols are the framework for exchange of data protocols. These are based on the PRIDE guidelines, or the promotion of research and innovation through data exchange guidelines, which are made by the DBT. So from morning, we heard various sessions, and we understood the importance of genomics data. And as you all know, because of ease of sequencing, we are generating more and more data worldwide. These data are primarily located in NCBI or EMBL or DDBJ. But if you see, India is not lagging behind, right? And this is because the NGS one can sequence. And if you look in terms of the number of databases in India, we are fourth in the world. So in terms of whenever we get the data, the genomics data or any kind of omics data, it becomes a responsibility to share it properly, right? And therefore there should be some guidelines. So the National Data Sharing and Accessibility Policy was formed in 2012, which was released by the Government of India. And later on, the Department of Biotechnology made the PRIDE guidelines for sharing the biotechnology guidelines with everyone. And this was released by Mantriji in 2021. So these guidelines are in harmonization with the international agreement and national policies, for example, Bermuda Accord, or for that matter, even the Indian policies like National Data Sharing and Accessibility Policy or Personal Data Protection Bill. So now question is, whose responsibility it is to share the data, right? So responsibility lies with all the stakeholders, whether they are funders, whether they are producers, where is the source, that means the individuals from where the sample is getting collected, or the users, or the custodians like IBDC or Indian Biological Data Center, where the Genome India data is
is kept, right? So it lies with everyone. And the feed protocols are the one which allows, right, to do this. And if you can see what it does basically, it allows the modalities and decision-making process. It's important that we have set guidelines to do this, and this conforming to the Biotech Pride Guidelines of 2021. And these guidelines, or these protocols, are binding on all the stakeholders. So that's important to understand that these feed protocols are binding to everyone. But in case there is a misalignment of national and international policies, then the National Act, rules, regulations, policies or guidelines, these will supersede, right? So PRIDE guidelines divide the data into 3 levels. Is the raw data, the processed data, and the metadata. And the data access levels are either open access or managed access or no access. So how these data will be shared? So there will be a DMG or data management group. They will review the access as per the Biotech Pride guidelines. And the decisions of the DMG will be reviewed by the EAC or the Expert Advisory Committee on a periodic basis. And if the data that is submitted to IBDC is a no access level, then the DMG and EAC will report this to Interministerial National Steering Committee. And it will go to them and they will take the decision on that. So these feed protocols basically define the responsibility of all the groups, whether they are data producers or submitters, or the custodians, or the users, or the data management group, or the funders. I will not go through the details of this, but the feed protocols defines the responsibility of each and everything which is related to the data, submission, or the user, or the management. And there are flow charts that have been made for the easy access of these data, and these are made for the users as well as those which are submitting the data, right? But this is quite complex, so let me simplify this for you. So let's say this is a managed access data, then the user registration happens. It is followed by the login and of course a data request. This is primarily first reviewed by the IBDC members, then it will be reviewed by IBDC head, and then this will be going to the data management group. They will make a decision on this, and then it will be shared with the user. So this is the sort of a flow process through which it will happen. Now, where is the data located? The Genome India data is located in Indian Biological Data Center, which is managed by RCB, or Regional Center for Biotechnology. So IBDC has a HPC, which is called BRUM, and that is where the data is. But the data is also, there is a recovery site in NIC Bhubaneswar, right? And IBDC not only hosts only the genomics data, but now it has various portals. For example, in terms of genomics, it hosts the open access data and the control access data, but it has crop phenome data, which is very unique to India. There are not too many crop phenome data overall in the world, and this is something which is very, very unique in India. We, of course, have the structural data and the proteomics data and metabolics data as well. All these portals are open, and the data is being submitted there as we speak. So as you can see that the data which is deposited is increasing, and from 23 to 24 there is a steep rise. Of course, that is also because of the Genome India data partly, but also we are getting more and more data. IBDC also conducts training, for various users as well as those who are submitters. And I would like to point it out particularly here. If you see that the usage of this data, IBDC, and you can see on your right that across India, people are actually submitting and as well as they are using data. So it's open, and if you have any issues, please contact us and we'll be more than happy to help you out. Thank you so much.
Thank you, sir. Now may I request Dr. Rajiv Bahel, Director General ICMR and Secretary DHR for his address.
Honorable Minister of Science, Shri Jitendra Singh Ji, respected Principal Scientific Advisor, Professor Ajay Sood, Secretary DBT, Dr. Rajesh Gokhale, Professor Narahari, Dr. Suchita, Dr. Sahu, and all the colleagues in this room. I am extremely delighted to be here to be part of this celebration of Indian science as a proud Indian, just like all of you. The importance of this work cannot be understated. Human genomics research is critical in improving health and healthcare. In particular, as was mentioned earlier, it is absolutely crucial for achieving the coveted goal of precision medicine, which we are moving towards. Genomics is making great strides in understanding complex conditions such as diabetes or heart disease, mental health disorders, or almost any condition from most common to the rarest of diseases. Therefore, Genome India, which is a unique data set in that it focuses on the diversity of our country by taking participants from all over India and giving the genome information, genetics information on all of them. I think it will greatly help us in understanding the genetic variations in our population, but also this data will provide a great opportunity for our not only academic and research institutions, but also for our industry to develop interventions which are diagnostics, therapeutics, or preventive interventions, thus integrating genomics into public health. So I would like to congratulate the Department of Biotechnology and all the scientists involved with this Genome India project for their wonderful accomplishment. We struggle and strive as scientists for years to have small moments of joy and celebration. So I join in really wishing them this wonderful on this success. They deserve a lot of congratulations, and I wish them success in using these data for improving the health and well-being of the citizens of our great country. Thank you, and Jai Hind.
Thank you, sir. May I now request Dr. Rajesh S. Gokhale, Secretary DBT and DG BRIC, to deliver his address.
Honorable Minister of Science and Technology, Dr. Jitendra Singh Ji, respected Principal Scientific Advisor to the Government of India, Professor Ajay Kumar Sood Ji, Director General ICMR and Secretary DHR, Dr. Rajiv Bahel Ji, Professor Y. Narahari Ji from CBR ISC, Dr. Arvind Sahu, Dr. Sucheta Ninave, who's coordinated this entire program, my colleagues from Department of Biotechnology, from institutions of DBT, various other scientific community, online and offline members, respected ladies and gentlemen. Namaskar and good afternoon to all of you. I think first and foremost, I would like to extend my heartfelt gratitude to our Honorable Prime Minister, Shri Narendra Modi Ji, for his very special video address that we will all have privilege to hear shortly after this. Your inspiring visions continue to motivate us to strive for excellence, and it is the honor to set the stage for your address, which will undoubtedly guide and inspire us to move forward in our mission. I would also like to sincerely thank Honorable Minister Dr. Jitendra Singh Ji. He has been a tremendous support, guidance, and his leadership has been absolutely outstanding and important for our own department's growth. I think its forward-thinking leadership is very unique. I must say this. I've had chance to now look at several other places, and that there's opened variety of vivid opportunities in the avenues of bioinnovations. Actually, he's particularly very keen on health perspectives and agriculture perspectives of human life, and I'm sure, sir, this will have a transformative effect on the futuristic growth in the impact of bioeconomy. I also like to thank our principal scientific advisor, actually, Professor Sood. Thank you very much, sir, for your guidance. You have been a great pillar of strength for us since you have been here. And I think thank you for accepting my last-minute request for you to join the meeting as well. I know you had a meeting, and thank you again. I also welcome my dear colleague, Dr. Rajiv Bahel. Thank you again for coming, and I think ICMR and DBT together can really make a transformative difference to the country's future in the world of medicine. I think today's event marked the landmark release of the Genome India data that symbolizes Bharat's scientific renaissance. For a very long period of time, we've been talking about our own genomic data, actually. We've had in bits and pieces and fragments everywhere, and we've been trying to scratch the data from different places to make it happen. And this is a real milestone for our country. It's a testament to our nation's renewed dedication to ingenious scientific growth, collaborations, more of the top of, and the creativity that will emerge out of this. As you would all realize, that genomic data acquisition now is the heart of almost all research-driven activities across various sectors. It's not only health. And this really sets us the standards for how we should manage and maintain our genomic data. The 10,000 genome program is absolutely outstanding in that way, is that we are now going to be able to release this data for every researcher working in every part of the world, not just in India, to be able to have an access to it, as Dr. Sahu so elegantly actually talked about it. I think I must congratulate and thank all the principal investigators. I think Dr. Narari, before that CBR head, Dr. Vijay Lakshmi, she was the one also taking care of these programs, actually, and the team members who are coordinators, contributors for exceptional, dedicated hard work. And I think the tremendous coordinated efforts has been the key to this success, actually. It truly reflects how complex challenges and problems can be solved by a collaborative networks, which is what Department of Biotechnology is actually more and more promoting, actually, to bring together consortiums of people to make sure we're able to take this, forte to the next generation level of activities. Of course, I need to thank my own DBT team, Dr. Suchita Nenave, and several other officers who, Dr. Narari already just talked about it, actually, for really pursuing this and taking this to the conclusive end, actually. Although this is just a beginning, I would again say. I mean, this, as you rightly heard in various discussions that you had prior to this, that this sets the standards, but it also makes us feel more ambitious to have 1 million or 10 million genomes of variety of kinds that will make us really proud for this country of small resource. I think one of the good things is that about 20 prominent institutions, whether they were from IISc, from IIT, from CSIR, from DBT BRIC institutions, they all came together to work on this platform. And I think that's where the real fun part of the game is, actually. And now the data is firmly secured at Regional Center of Biotechnologies, IBDC, with the Indian Biological Data Center that provides a wonderful resource for all of us to access. And my request to all of you is that every data that you generate, which you submit at NCBI for all the other purposes, please submit it as IBDC as well, actually. I think the more data we will submit, the more resourceful it will become. The team is already developing various creative programs and softwares on top of it that will be enormously enriching. And the primary data being in our country would be a transformative for AI-driven work that is likely to make sure the future's generation work. Of course, as we start using this data, we have to also be assured of how we make use of this data, actually, and what kind of valuable protocols and the framework of exchange of data protocol that you talked about actually sets now an access for what we call fair, transparent, and open access to the biological data while ensuring the ethical standards of protecting participants' privacy and fostering trust in scientific research. And I think that's all very, very important given that we actually live in this very complex world. I must say the Department of Biotechnology remains committed to delivering several such important events as we move along. And as I call the acronym DBT as delivering a better tomorrow, we are likely to support several new endeavors. For example, bioeconomy and biomanufacturing is going to be immensely benefited by this baseline leveraging data in a variety of ways. As my colleague from ICMR DG said, the precision medicine cannot happen without this. And I think what all we're looking for future of medicine is going to be precision medicine of how we specifically identify diseases, how we specifically cure them, and how we provide protocols that are better for the person's individual perspective. It also provides efficient production of biopharmaceuticals, variety of other activities that will happen around this actually. Moreover, the insight derived from the data can stimulate biotech startups. It can provide creative value of bioproducts while reducing dependency on traditional manufacturing products. The boost global competitiveness, and we hope that this will create new jobs and new activities as well, because as people endeavor into doing data analysis and activities, it will be wonderful opportunity for obtaining sustainability models of governance. The journey underscores the nation's strategic transition from being a hub of generic manufacturing to a global leader in biomanufacturing policy and policies like BioE3, which positions biotechnology as a key driver of economic, employment, and environment goals are pivotal to this transformation. Let us continue to build this momentum we have created, and I once again commend this extraordinary scientific team efforts for their success and dedication. Together we will achieve many more milestones and contribute to our shared goals for a brighter and more innovative future. Thank you. Namaskar.
Thank you, sir, again for requesting everyone for data sharing, which is of prime importance for acquiring the maximum benefit from public funded research. Now may I request Professor Ajay Kumar Sood, Principal Scientific Advisor to the Government of India, for his kind address.
I'm absolutely delighted to join the galaxy of dignitaries on the dais, Honorable Minister Dr. Jitendra Singh Ji, Dr. Rajiv Behal, Dr. Rajesh Gokhale, Professor Narahari from my parent institute, and Dr. Suchita, Dr. Arvind, and all of you enthusiasts in biotechnology, biology, and all the related subjects. It's indeed a great moment, as somebody, I think Narari said, watershed moment, indeed, to have the release of this Genome India data, but also a wonderful example of collaboration across the institutes along with the government support. I think this unique blend of bringing all the partners together, which is happening more and more in the last few years, this is one more example of the strength of this collaboration, which can deliver much more results with our efforts. Now, needless to say to this audience that the journey of science and technology in revolutionizing healthcare is a tale of unprecedented progress. In the last almost 75 years, I vividly recall, on a personal note, reading the book The Double Helix by James Watson when we were growing up. It was a fascinating journey, even the stories in that, how the discovery was made, how there was a competition to discover this double helix structure. I still remember in my student days reading that and discussing among ourselves. And I also recall when I came to do BSc and MSc, reading that paper again, and there was a very beautiful note in the last paragraph, which I took so much note because these days it is so important. All of us, when we write our papers, we say it's the first time we have discovered it. It is this, that. But in that paper, which is so fundamental, it made a very nice line, and I would like to read for you. It says, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. I don't think it could have been more prophetic, but it was so humbly put, example for all of us when we go overboard in making a small science results into larger than size. So this is really humbling. And at this moment, I thought I'll recall my impressions of those days when I read that paper. So we all know that we have come a long way with the discovery of DNA, and Genomic has greatly enhanced healthcare by introducing personalized medicine, as was mentioned before, and transforming the diagnosis of genetic disorders. And this has shifted healthcare from one size fits all model to a more individualized approach. This is already happening in various countries where they are having a genomic database like US, UK, Japan, and so proud that India has joined this with this first set of 10,000 genome sequences, and already promised by Dr. Rajesh Gokhale, and we take it very seriously, whatever is said on stage, that it will be 1 million soon. So you can't go away from that promise. So, recorded. And if it is not done, it is not good for you. So you have to deliver now 1 million thing. But I'm saying this is what is called aspiration. You can aspire now, which was not happening earlier. And I'm extremely confident that this will happen by this collaborative effort of academia, government, and industry. We need to bring more and more industries into the fold, and then this 1 million does not look a very dreamy target. It can be done, and compliments to Rajesh for thinking of this and really putting his dream on the dais for all of us to work for it. And for a country like India, we all should remember we are almost 1/6 of the globe, 1/6 on Mother Earth, with diverse demographies. The genetic makeup of this our citizens is extremely crucial, and whatever was said in the introduction by Rajesh and other speakers, we all know how it is important for tailoring the public health policies, and for that, absolutely congratulations to the leadership of DBT, congratulations to the leadership of the country, Dr. Jitendra Singh ji, for supporting this, and to our Honorable Prime Minister, who has led from the front in taking this technology approach to look at the societal problems in no uncertain ways, when he also introduced Anusandhan as the fourth dictum along with the other three from the Red Fort. I don't think it could have been emphasized in a better way. And we are all soldiers in that army to fulfill the dreams of taking this Anusandhan to the societal goals. And this genome sequence, the baseline data which has come up in these studies, will really serve a very, very decisive role. And this will help. I don't want to belabor how it will look at the very important problems like cardiovascular and cancer cases which are rising. I mean, the numbers are frightening in cancer when I read in the literature, and I'm very sure cancer research will take a very different turn when we bring the whole genomics into it. And this is, I understand it's happening, but it should happen in our country in a way that we can be the leaders in the world. So this dedicated genomics repository, needless to re-emphasize, will boost our research and innovation and bring advancements in personalized medicine and enhancing national security by reducing reliance on the foreign data, which is what we have been using, and we know it is not the best thing because our demography is different, our genomic makeup is different, and so on. So here I just want to mention two more missions with the, I hope I am not exceeding my time, but just I cannot avoid mentioning two important missions which will have direct influence on this genomic sequence. And that is the National AI Mission, which was launched in March 2024 with $1.3 billion investment by Government of India, almost like 11,000 crores, and the National Quantum Mission, again launched in 2024. So why am I mentioning again quantum? Because the conventional way of doing is the AI is doing by the classical computers using bits. But when the data becomes enormous, I don't think it has escaped people's attention that quantum computing is the future to look at the genome sequence along with other problems like climate change, weather prediction, all that. But genome sequencing, understanding genome in quantum computing is a paradigm which I'm sure will be a reality in coming years. So this marrying the other two missions of what was announced by government and this Genome India initiative can really transform this whole approach to this biotechnology. So and AI, needless to say, is a game changer, which all of us are absolutely convinced that the way to go. So I think I will only mention one more thing from the point of view of this genome thing. We have the digital twin concept already for many years now, and now we are also having a concept of digital genetic twin, which is what the digital doppelganger. And this is what I think we should be also thinking right away in the research domain. How by leveraging insights from genomic data set, we can create a digital genetic twin. And this, which is a virtual replica of individuals' biological makeup. And using advanced AI and machine learning algorithms, this digital twin will continuously update with new data and reflecting changes in the individual's health status over time. So this innovative approach, I am sure, will highly personalized medicine with treatments and so on. And I look forward to really get to know more about this digital genetic twin in coming months and years. I am quite a fan of this digital twin, AR, VR, and all the new technologies. And this digital genetic twin, I have not heard much in India, but I hope that with the DBT and ICMR support and our leadership support, we would be making leadership role in this upcoming technology in the next 5 years. So it all looks excellent. We are absolutely delighted. And the proposed framework which was presented by Dr. Arvind on this FEED protocols, again, is very good to, it assures you that it is in good hands. And there are protocols which will be followed, and there are safety issues which have already been put in place, which is so very essential in the days of cyber security and all the threats from our adversaries and so on. So once again, I would like to congratulate DBT for supporting this. All the team, we have representation by Professor Narhari and his entire team. I know it's not one person's effort. It is a collective effort, and our leadership who really supported all this, and all of you who are partners in this journey. Thank you very much.
So thank you for your encouraging words. May I now request the Honorable Minister of Science and Technology, Shri Jitendra Singh Ji, for release of the framework for exchange of data protocols. May I request Dr. Nair and Dr. Richiva Ajan to join on the dais. I request the Honorable Minister to launch the IBDC portal for data access. I again request the Honorable Minister to launch a call for proposal on the Genome India dataset. Thank you, sir. Now may I request Dr. Jitendra Singh Ji, Honorable Minister for Science and Technology, to address the August gathering. Thank you, sir.
Principal Scientific Advisor to the Government of India, Professor Ajay Sood, Secretary, Department of Biotechnology, Dr. Rajesh Gokhale, Director General, ICMR, Dr. Rajiv Bahel, Dr. Narahari, Dr. Sahu, Dr. Suchita, representatives of the different stakeholder agencies, institutions, members of the fraternity, ladies and gentlemen. I think as was mentioned by Dr. Rajesh Bahel and then also by Professor Sood, this is indeed an occasion to celebrate, and a celebration which would be realized more by those who are involved in it rather than others, because a subject happens to be relatively hitherto not fully explored. But nevertheless, I think I would pick up from what Professor Sood was saying, that the celebration has to be also quite modest, because we are still learning, and the more modest we are, the better learners we would be. And when he was referring to that book which he read as a student, I'm sure Dr. Bahel at least would bear me out that when we were students, we had in pathology the famous book by Boyd's, because later on, of course, the entire science was taken up by the Americans, and most of the books were by the British authors. So that was considered to be the ultimate Bible in pathology. So it's a voluminous book, and the last sentence in the preface that Dr. Boyd wrote was that ultimately I have also to thank the girl who was doing my drafts for taking my last minute changes with apparent magnanimity. So sometimes you also tend to learn from the person who is taking down your notes, though he may not be actually a researcher, but having been associated with you, then trying to pick up from what you're writing, sometimes he gets also sometimes an instinctive sense of the word sometimes used in some contexts. So I think it's a very modest team effort, and we have actually begun with a breakthrough. And he was saying that every paper writes that. I think there's a huge influence of the television happening, the breaking news actually echoing in the breaking research. But most of us from the fraternity who have walked a long way have not succumbed to that temptation. But if you see in a total perspective, I think the last 1 decade has been a game changer for India's biotechnology in more ways than one. If you see statistically, because we've been taught to speak through figures and statistics to make evidence of what we are saying, we were just about 50 startups in 2014. Now we've gone up to almost 9,000. We were just about $10 billion economy, went up to 80 in just 8 years, now somewhere close to 130, 40, and looking forward to be 300 billion in the next few years. In the biomanufacturing, we are ranked number 3 in the India Pacific and somewhere around 12, among the first 12 in the world. And I think we had an opportunity of proving our acumen through the language of vaccine. Now, COVID came as a pandemic, as an adversity, but every adversity is accompanied with a virtue. So it came as a virtue for biotechnology, though I don't wish that it happens again and again. But it gave us an opportunity to prove to the world that we could come out with the first ever DNA vaccine. And to my mind, why it makes the rest of the world startled is that the world, particularly the West, never took us seriously as far as this was concerned. We were never taken a nation, even at the society level, very serious, even about the therapeutic healthcare. If you fell sick, you waited for 3-4 days before deciding to consult a clinician or a physician. You waited for some divine powers to come to your healing. कोई माँ का आशीर्वाद आ जाएगा, prognosis खराब हो गया तो पिछले जन्म का बुरा कर्म होगा. So many extra clinical remedies also got jungled up. So a country like that, a nation like that, because I remember when many years ago one of the health ministers, because it's only now in the last 10 years that I've started sitting here, otherwise I used to sit on that side and watched series of health ministers passing, because I've never been a part of the government setup. So one of the health ministers who said, in my village, if anybody died, they would rather arrange prayers and say कि डायन आ गई थी, अब किसी और को न लग जाए. She used to tell about some remote village in Uttar Pradesh. So for a country where even the therapeutic healthcare was not a priority, leave aside at the researchers level, even in the government level, the society level, that country today is being acknowledged as a leader in preventive healthcare. So that is something remarkable. That is what has taken aback the world. And this is not a fluke. Now we have come with the papillovirus vaccine, and then series of others already in the pipeline. And then in the context of the subject today, the gene therapy trial in hemophilia conducted in Christian Medical College, Vellore, has found mention, in fact a whole article in New England Journal of Medicine. Now sometimes we researchers and scientists take pleasure in saying कि हमारे journal कौन से कम है. I'm sure Professor Sood, because he's a teacher of teachers and I've been a teacher of sort also, I for one take it as an apology, because you don't get it published and you say so. It's not, the question is that, I mean, you could be a nationalist, but it's not the question of being less nationalist by finding acknowledgement by a recognized body. So now New England Journal, why I'm saying so to this audience who are already part of the fraternity is almost 200 years old. And it belongs to the times when entire science was controlled by the United Kingdom, which is a larger Commonwealth world. Americans took over somewhere in the middle of the 20th century. And New England Journal of Medicine and the British Medical Journal, Dr. Behel would bear out, were the two premier journals came out of London. And if that journal publishes our trial, a trial which was led by the team headed by Dr. Rajesh Gokhale, means that they have vetted it through all possible parameters, and then they own the responsibility of telling the world that, look here, this is an authentic trial happened. It's not the question of the foreign journalists, but I'm saying, so that has actually given us recognition, acknowledgement, and made the world serious about the kind of work that we are doing. And most of it, as Rajesh was rightly mentioning, has been possible because we have a political dispensation headed by Prime Minister Modi, who is forthcoming, who is very intelligent, very supportive. I sometimes tell myself, because I was with Rajesh when they were preparing these Bio E3 presentations again and again and again and again, going back to convince everybody, I sometimes tell myself that if we didn't have Prime Minister Modi, if we didn't have Mr. Modi as Prime Minister, we wouldn't have been able to get this pass. It happened because, I mean, there were so many others kept saying, why are you indulging in this? What's the hurry? What's going to happen? So in repeated representations, we could explain to him that, look, sir, this is going to be the next phase, the bioeconomy, and the bioeconomy is going to take over in a big way. And we, with our sources, huge bioresources, capabilities of regenerative processes, replacing the manufacturing economy, we could at least be into it and tell the world that we are already into it. And finally, he accepted it, and Rajesh was, who had worked hard with the president. So I think that kind of support, encouragement has given us confidence to move on. That political patronage, which perhaps was not forthcoming in the same measure before 2014. And that has happened. I don't know whether that's happened to you, because this occurred to you, because that has a political connotation. This policy was among the highlights of the first 100 days of Modi government 3.0. I mean, this came as a priority soon after the third term started. And the other highlight of the first 100 days of Modi government 3.0 was allocation of 1 lakh crore for National Research Foundation, Anusandhan. Now, if that is the kind of prioritization in the political government, I think that speaks for itself with enough evidence. One more, because I wish that we go out and tell the people about this. You remember we had this vote of accounts. Whenever a government is going for election, they don't come out with a budget, with the sacrosanct spirit of democracy that the budget priorities are the prerogative of the incoming government. So I leave that in the true spirit of democracy to the next government after the election which takes over. So, but Kaam Kaji Kharcha is vote of accounts. In that vote of accounts, we got two mentions. One was 1,000 crore for space startups, and the other was semiconductors. In fact, even I didn't expect that. I went around telling others, maybe others didn't realize it. Usually, vote of account consists of free laptops and sewing machines. So government going in for election is talking of semiconductors at the startups and space sector. So that is the kind of conviction with this government headed by Prime Minister Modi has worked and given us a different kind of culture. That's what has made it possible. And of course, as Professor Sood was saying, this is one of the beautiful illustrations of the whole of government approach happening. We have the ICMR, we have the other institutions, and I was telling Rajesh that we could also rope in the Tata Memorial, because as was being mentioned, that we don't get enough of data from the cancer institutes. So we have other one or two, the private ones are also doing, like for example, I believe one or two institutes have established this SOP of going in for genetic studies whenever there are more than one members of a family having malignancy. But they don't come out with data, or they don't bother to provide us with, to add to our data bank, as was Rajesh complaining. But I think Tata we can motivate it because it's functioning under the aegis of the Department of Atomic Energy, and we can request the Secretary Department of Atomic Energy, which also comes under the purview of the PSA's office. So we could also ask them to be a part of this effort. And a larger ecosystem which is now developing, and as Professor Sood rightly said, the artificial intelligence and the quantum are going to actually facilitate this. Now, as far as these two releases this evening are concerned, genes, as I said, is a relatively undervisited, or for many an unvisited area. So that makes it little exclusive, little distinct. And I, for one, and many of me belong to the generation we have seen this transition happening. Like, movies starting from the child stars, then growing up, then singing a duet, recalling what was happening in the childhood, and then growing old, and then happily walking out at the end before the national anthem. When we started off in our third year, we had a very meticulous class of prescription writing, which doesn't happen now. If you tell the students now, they laugh at us. So they would tell us how to write the prescription for preparing a cough mixture, for preparing a diarrhea mixture, because pharmacology was in a very, very primitive state. And then gradually, the advent of antibiotics. We used to have a posting just to learn injection. That was in third year. I remember the first injection I gave, the person collapsed. But the nurses were so generous. She said, no, nothing happened. Just give him this. So she immediately put in a steroid injection and reported, because he had actually reacted to the test dose of penicillin. So all this was considered to be a skill and an art to be trained for, which no longer exists now. We've gone that far. When we got into post-graduation, the first day, your MD guide would tell you to get a retinoscope or a fundoscope, because you were meant to see the fundus, because whether it's hypertension, it's diabetes, it's malignancy, everything would reflect on that. Now the ophthalmologists don't use this, because they place the face before the screen, and what you get through artificial intelligence is much better than the subject. So this transition has happened, and therefore gene therapy and all this gene-related options are going to be an essential part of the armamentarium in our future diagnostic as well as management techniques. And therefore, we need to, that brings the necessity of, and this is what I think we need to have also programs among the students, also in the public, because of course in our conferences we started having two sets of it, the public speeches as well as the academic speeches. We need to, because therein where we need Indian data. And as I always been saying for years, Indian solutions for Indian problems, Indian remedies for Indian diseases. And as Professor Sahu mentioned that for 140 crore population they have gathered, and there are more than 4,600 demographic groups, and sequencing 10,000 individuals from different communities, as many as 100 or 99 communities, is no small task. But of course, the more larger and more representative would be the data. And therefore, we are aiming at 1 million, which would also be an exercise to map out the disease vulnerability, the disease susceptibility, the propensity, because India is such a varied society and a community, even genetically, unlike many of the European countries. There's no homogeneity at all. And it's been now understood that even a particular gene may behave differently in different communities. Like, for example, the atherosclerosis is more common, a pathogenic component for coronary artery disease amongst us, among the Southeast Asians. Whereas in the Africans, it is more in the cerebrovascular, causing more of brain strokes. So the same predisposing factor producing different outcomes because of the genetic variations. Our Indian phenotype, even Indians having settled in Europe for 3 generations continue to have a higher propensity of certain diseases like type 2 diabetes mellitus, and even the central obesity. Only I think doing injustice to, so we appointed another team, obviously he is also. And we found that now the central obesity is equal in males and females almost in India, almost 63-64%. And it's known to be an independent risk factor for all these metabolic disorders, independent and regardless of the overall obesity, which is the reason why the Westerners appear to be obese and fat, but their visceral fat is very little. Unlike us, we wear a long kurta, wear a long sari, it gets camouflaged. So all these things, so I think now the answers are going to be discovered through these data, and at the same time, the concerns about confidentiality were raised even when we started this exercise, and I'm glad I must compliment Dr. Sahu and the team for this framework for data exchange. Now, the point is that you want to exchange and yet not want to exchange. And then the dilemma is how much to exchange, how much not to exchange. So to decide the optimum of it is a great challenge. But without it also you can't move on. And therefore, this is an equal, I think, as part of the ecosystem, and particularly for the interdisciplinary collaborations. And I'm sure this, the exercise accomplished today will be a step forward in our world arena competitiveness in drug development, in personalized treatment of disease, which is something I think going to happen, I don't know whether in our lifetimes or beyond, because for the same disease you may not be amenable to the similar kind of treatment. And that will be determined by the nature of your gene or by the behavior of your gene. And then, of course, the breakthrough in the different diseases, like we talked about hemophilia, and of course the rising prevalence of malignancies and type 2 diabetes mellitus in this country. And last but not least, as I also mentioned, how to create more of public awareness about it. That's very important, because stakeholders, we have whole of science, stakeholders are not confined only to the scientific fraternity. Ultimately, you are targeting a common citizen, regardless of his scientific acumen, scientific qualification, scientific knowledge. And your ultimate, in fact, in our times when the clinical medicine got more of supremacy compared to textbooks, we were taught the patient is the best teacher. So ultimately your target is the individual. So unless you make him a stakeholder and you bring in pressures, like as Secretary DBT was rightly mentioning, the constraint of procuring data or not being shared, when we start getting pressure for the social groups, look here, for the benefit of all, like it happened in COVID. There was pressure coming in from the other groups as well, from those who didn't have a direct stake in research. So we need to create awareness about the importance of what is being done. Otherwise, to a common people, it appears to be something very vague. They feel we are sitting in a dark room, in a dark lab, doing something just to get a month's salary. They don't realize that what is being done has a very, very close bearing on their future existence. And therefore, I think we will devise a method of that through the social media options, and also by arranging some kind of a public outreach program. But overall, whether today we realize or not, I think today is a day which marks a frontal leap for India as far as our excellence in the global role that we are envisaging for ourselves, and also for a bioeconomy-driven fourth industrial revolution, because we have the advantage of that. The bioeconomy-driven India's contribution would be based on our biodiversity, our biofoundries, our genetic data. And therefore, to that extent, in the coming times, it may not be realized that deeply today, it is this group of persons, people sitting in this room, who would, to an extent, get the credit of having contributed to the making of Viksit Bharat. It may not be realized today, but I'm sure in 2047, when they who celebrate 2047 look back, they would also look back to the contribution made by this team. Well, this is the future for the world, and India has already taken a lead. Thank you.
Thank you, sir, for the inspiring words and providing valuable insights into government's vision for coming times. Prime Minister Narendra Modi, Honorable Prime Minister Shri Narendra Modi's commitment to science is evident in various initiatives that focus on innovation, research, and technological advancements. The video address will announce the release of this landmark dataset.
केंद्रीय मंत्रिमंडल में मेरे सहयोगी डॉक्टर जितेंद्र सिंह जी, देश भर से यहां आए सभी साइंटिस्ट, अन्य महानुभाव, देवी और सज्जनों, आज भारत ने रिसर्च की दुनिया में बहुत ही ऐतिहासिक कदम उठाया है। 5 साल पहले जीनोम इंडिया प्रोजेक्ट को स्वीकृत किया गया था। इस बीच कोविड की चुनौतियों के बावजूद हमारे वैज्ञानिकों ने बहुत मेहनत से इस प्रोजेक्ट को पूरा किया है। मुझे खुशी है कि देश के 20 से ज्यादा दिग्गज रिसर्च संस्थानों जैसे IISc, IITs, CSIR एंड BRIC ने इस रिसर्च में अहम भूमिका निभाई है। इस प्रोजेक्ट का डेटा 10,000 भारतीयों का जीनोम सीक्वेंस अब इंडियन बायोलॉजिकल डेटा सेंटर में उपलब्ध है। मुझे विश्वास है बायोटेक्नोलॉजी रिसर्च के क्षेत्र में ये प्रोजेक्ट एक मील का पत्थर साबित होगा। मैं इस प्रोजेक्ट से जुड़े सभी साथियों को बहुत बहुत शुभकामनाएं देता हूं। साथियों, जीनोम इंडिया प्रोजेक्ट भारत की बायोटेक्नोलॉजी रिवॉल्यूशन का एक महत्वपूर्ण पड़ाव है। मुझे बताया गया है कि इस प्रोजेक्ट की मदद से हम देश में एक डाइवर्स जेनेटिक रिसोर्स बनाने में सफल हुए हैं। इस प्रोजेक्ट के तहत देश में अलग अलग आबादियों से जुड़े 10,000 लोगों की जीनोम सीक्वेंसिंग की गई है। अब ये डेटा हमारे वैज्ञानिकों को, रिसर्चर्स को उपलब्ध होने जा रहा है। इससे हमारे स्कॉलर्स को, हमारे साइंटिस्ट को भारत का जेनेटिक लैंडस्केप समझने में बहुत बड़ी मदद मिलेगी। इन जानकारियों से देश के नीति निर्धारण और योजनाओं के निर्माण का काम भी आसान होगा। साथियों, आप सभी यहां अपनी फील्ड के एक्सपर्ट्स हैं, बड़े वैज्ञानिक हैं। आप भी जानते हैं भारत की विशालता और भारत की विविधता सिर्फ खान पान, बोलचाल और भूगोल की ही तक सीमित नहीं है। भारत में रहने वाले लोगों के जो जीन्स हैं उनमें भी काफी विविधता है। ऐसे में बीमारियों का नेचर भी स्वाभाविक रूप से विविधता से भरा हुआ है। इसलिए कौन से व्यक्ति को किस प्रकार की दवा फायदा देगी, ये जानना बहुत आवश्यक है। इसके लिए देशवासियों की जेनेटिक आइडेंटिटी, उसका पता होना जरूरी है। अब जैसे हमारे आदिवासी समाज में सिकल सेल एनीमिया की बीमारी एक बहुत बड़ा संकट है। इससे निपटने के लिए हमने नेशनल मिशन चलाया है, लेकिन इसमें भी चुनौतियां कम नहीं है। संभव है कि सिकल सेल की जो समस्या किसी एक क्षेत्र में हमारे आदिवासी समाज में हो, वो दूसरे क्षेत्र के आदिवासी समाज में ना भी हो, वहां दूसरे प्रकार का हो। इन सारी बातों का पक्का पता हमें तब चलेगा जब एक कंप्लीट जेनेटिक स्टडी हमारे पास होगी। भारतीय आबादी के अनूठे जीनोमिक पैटर्न्स को समझने में इससे मदद मिलेगी और तभी हम किसी खास ग्रुप की विशेष परेशानी के लिए वैसे ही विशेष सॉल्यूशन या फिर प्रभावी दवाएं तैयार कर सकते हैं। मैंने सिकल सेल का उदाहरण दिया है, लेकिन ये इतने तक सीमित नहीं है। ये तो मैंने एक उदाहरण के लिए बताया। भारत में आनुवंशिक रोगों यानी एक पीढ़ी से दूसरी पीढ़ी को ट्रांसफर होने वाली बीमारियों के बहुत बड़े हिस्से से आज भी हम अनजान है। जीनोम इंडिया प्रोजेक्ट भारत में ऐसी सभी बीमारियों के लिए प्रभावी इलाज के विकास में मदद करेगा। साथियों, 21वीं सदी में बायोटेक्नोलॉजी और बायोमास का कॉम्बिनेशन बायो इकोनॉमी के रूप में विकसित भारत की बुनियाद का अहम हिस्सा है। बायो इकोनॉमी का लक्ष्य होता है नेचुरल रिसोर्सेज का सही इस्तेमाल, बायो बेस्ड प्रोडक्ट्स और सर्विसेज का प्रमोशन और इस सेक्टर में रोजगार के नए मौके बनाना। बायो इकोनॉमी सस्टेनेबल डेवलपमेंट को गति देती है, इनोवेशन को अवसर देती है। मुझे खुशी है कि बीते 10 वर्षों में देश की बायो इकोनॉमी तेजी से आगे बढ़ी है। साल 2014 में जो बायो इकोनॉमी $10 बिलियन की थी, वो आज $150 बिलियन से ज्यादा की हो गई है। भारत अपनी बायो इकोनॉमी को नई बुलंदी देने में भी जुटा है। कुछ समय पहले ही भारत ने बायो E3 पॉलिसी की शुरुआत की है। इस पॉलिसी का विजन ये है कि भारत आईटी रिवॉल्यूशन की तरह ग्लोबल बायोटेक लैंडस्केप में भी एक लीडर बनकर उभरे, इसमें आप सभी वैज्ञानिकों की बड़ी भूमिका है और इसके लिए मैं आपको अपनी शुभकामनाएं देता हूं। साथियों, विश्व के एक बड़े फार्मा हब के रूप में भारत ने जो पहचान बनाई है, उसे आज देश नया आयाम दे रहा है। बीते दशक में भारत ने पब्लिक हेल्थ केयर को लेकर अनेक क्रांतिकारी कदम उठाए हैं। करोड़ों भारतवासियों को मुफ्त इलाज की सुविधा हो, जन औषधि केंद्रों में 80% डिस्काउंट में दवाएं उपलब्ध कराना हो, आधुनिक मेडिकल इंफ्रास्ट्रक्चर का निर्माण हो, ये पिछले 10 साल की बहुत बड़ी उपलब्धियां हैं। कोरोना काल में भारत ने ये सिद्ध किया है कि हमारा फार्मा इकोसिस्टम कितना सामर्थ्यवान है। दवाओं की मैन्युफैक्चरिंग के लिए भारत में ही मजबूत सप्लाई और वैल्यू चेन बने, ये हमारा प्रयास है। जीनोम इंडिया प्रोजेक्ट अब इस दिशा में भारत के प्रयासों को नई गति देगा, नई ऊर्जा से भरेगा। साथियों, आज दुनिया ग्लोबल प्रॉब्लम्स के सॉल्यूशंस के लिए भारत की तरफ देख रही है। हमारे आने वाली पीढ़ी के लिए एक रिस्पॉन्सिबिलिटी भी है, एक अपॉर्चुनिटी भी है। इसलिए आज भारत में एक बहुत बड़े रिसर्च इकोसिस्टम का निर्माण किया जा रहा है। बीते 10 सालों में रिसर्च और इनोवेशन के लिए पढ़ाई के हर स्तर पर बहुत जोर दिया गया है। आज 10,000 से ज्यादा अटल टिंकरिंग लैब में हमारे स्टूडेंट्स हर रोज नए नए एक्सपेरिमेंट कर रहे हैं। नौजवानों के इनोवेटिव आइडियाज को आगे बढ़ाने के लिए पूरे देश में सैकड़ों अटल इंक्यूबेशन सेंटर बने हैं। पीएचडी के दौरान रिसर्च के लिए पीएम रिसर्च फेलोशिप स्कीम भी चलाई जा रही है। मल्टी डिसिप्लिनरी और इंटरनेशनल रिसर्च को बढ़ावा मिले, इसके लिए नेशनल रिसर्च फंड बनाया गया है। अनुसंधान नेशनल रिसर्च फाउंडेशन में, उससे देश में साइंस, इंजीनियरिंग, एनवायरमेंट, हेल्थ ऐसे हर सेक्टर में और नई प्रगति होने वाली है। सनराइज टेक्नोलॉजीज में रिसर्च और इन्वेस्टमेंट बढ़ाने के लिए सरकार ने 1 लाख करोड़ रुपए का कॉर्पस क्रिएट करने का भी निर्णय लिया है। इससे बायोटेक्नोलॉजी सेक्टर का भी विकास होगा और यंग साइंटिस्ट को बहुत मदद मिलेगी। साथियों, हाल ही में सरकार ने वन नेशन वन सब्सक्रिप्शन का एक और अहम फैसला लिया है। दुनिया के प्रतिष्ठित जर्नल्स तक भारत के स्टूडेंट्स की, रिसर्चर्स की पहुंच आसान हो, उन्हें खर्च न करना पड़े, हमारी सरकार ये सुनिश्चित करेगी। ये सारे प्रयास भारत को 21वीं सदी की दुनिया का नॉलेज हब, इनोवेशन हब बनाने में बहुत मदद करेंगे। साथियों, जिस तरह हमारे प्रो पीपल गवर्नेंस ने, हमारे डिजिटल पब्लिक इंफ्रास्ट्रक्चर ने दुनिया को एक नया मॉडल दिया है, मुझे विश्वास है उसी तरह जीनोम इंडिया प्रोजेक्ट भी जेनेटिक रिसर्च के क्षेत्र में भारत की छवि को और सशक्त करेगा। एक बार फिर आप सभी को जीनोम इंडिया की सफलता के लिए मेरी ढेर सारी शुभकामनाएं। धन्यवाद। नमस्कार।
We are indebted to Honorable Prime Minister Shri Narendra Modi for his kind words and highlighting the government's supports for biotechnology, space exploration, health research, agriculture, which illustrates a holistic approach to integrating science into national development. Before we close this session, it is my honor and privilege to stand before you to express the deepest gratitude on behalf of the Department of Biotechnology as we conclude today the Genomics Data Conclave. At the outset, I would like to extend our heartfelt thanks to Honorable Prime Minister Shri Narendra Modi ji for graciously sparing valuable time to address this gathering through the video message, despite numerous other commitments. His efforts to connect with us today speaks volumes about his unwavering dedication, commitment to scientific temperament, and technological innovation in charting new territories for country's progress. His leadership has driven by a deep sense of purpose, which continues to inspire us all, and we are fortunate to be guided by a leader who not only articulates a compelling vision, but also embodies the principle of hard work, perseverance, and resilience. Working in tandem with Honorable Prime Minister in realizing the vision of Atmanirbhar Bharat, Honorable Minister Shri Jitendra Singh, Honorable Minister of Science and Technology Shri Jitendra Singh ji has been guiding force for the department. We extend deepest gratitude for his unstinted support and not only motivating us but empowering us to believe in future and which we can collectively build. Sir, the vision you have shared today is just not the roadmap for progress. It's a call for action that compels us, each of us, to contribute with renewed dedication and purpose. We are immensely thankful to Professor Ajay Kumar Sood, PSA to Government of India, for gracing this occasion with your presence and also for inspiring the address that undoubtedly enriched our perspectives. I would also extend my sincere thanks to Dr. Rajesh S. Gokhale, Secretary DBT and DG BRIC for his invaluable mentorship, unwavering support, and thought-provoking perspectives. Thank you, sir, for the personal example you set for all of us and instilling in us the confidence to work together as a team for achieving the goals set forth. I would also like to express my sincere thanks to Dr. Rajiv Bhal, Secretary, Department of Health Research and DG ICMR, for joining us in this data genomics conclave and giving insightful viewpoint on leveraging the genomics data for further health goals in the country. I wish to place on record my heartfelt thanks to Dr. Suchita Ninave for steering the Genome India project in seamless manner and coordination of this event and maximizing the outcomes of the program through her untiring efforts and unflinching support for all stakeholders. I would also like to express sincere thanks to all the distinguished dignitaries, panelists, speakers who have taken time to join us today. Besides contributing significantly to the success of this event, they have also presented that this research fraternity presents the new avenues and endless prospects for utilizing the data in solving critical problems. Our gratitude extends to the participants of the Genome India team who tirelessly worked out for the last 5 years for accomplishment of this project. I also wish to thank the members of Data Management Group, Expert Advisory Committee, and National Steering Interministerial Committee for drafting and shaping of feed protocols. I would also like to acknowledge the tireless efforts of organizing committee and all the individuals behind the scenes. Without your dedication and hard work, this event would have not been possible. Just to mention, this has been hosted by RCB and IBDC teams, so this event would have not been possible. We truly appreciate your commitment to ensuring the smooth execution of this gathering. In the last, I would like to thank the senior officials of Ministry, members of press, and all other dignitaries who are present online for joining us on this Genomics Data Conclave. Thank you. We have a tea arranged in the atrium area. Thank you.